Good day, everyone. My name is Peter Pappas. I'm with the MSGERC, and we're doing something today that we've not done before, and that is that we're having a joint enterprise, joint session with selected members of the Niad with a focus on the bacteriology and mycology branch. I won't go into the details as to how this came about, but the short story is that we were exploring some options or exploring some ideas with Donna Love and Bang Liu, who you're going to meet and hear from very shortly in January, and they were expressing concern that they weren't seeing a lot of clinical and translational proposals through their section that with a focus on mycology. And since that is sort of their thing, we thought we'd like to try and address this by providing information, starting from the inside, that is, starting from the intramural folks, those who are at the agency itself, and then intramural investigators as well, get their perspective on working, within the NIH or working within the NIAID. And then we're going to get this perspective of those who are extramural funded investigators who've had success with their applications to NIAID and other agencies for that matter, but especially through NIAID, to get their perspective on how this has helped them, how they've been successful, insights that they can provide us that will allow those of us in attendance to maybe get some ideas, think a little bit differently and more creatively about how we go about doing this. And lastly today, we are going to hear from several young investigators who are basically going to float some ideas, some brief proposals with the purpose of getting some feedback, some immediate feedback from everyone who's in attendance today. So I'm not going to spend a lot more time on this. I'm going to get started here with the presentations real shortly. I would like to thank those who put together the details of the conference today at Caslin and Associates. I'd like to thank Carolyn Jones, our administrative director of the MSGERC, who've basically gotten the program really in order. And I'd like to thank all the participants today, those of you who've volunteered to provide your insights. I want to indicate that questions will be answered at the end of each session. You can use the chat or the raise hand function. Pardon me. And with that, I'm going to introduce the first two speakers, and then they will in turn, I'm not sure who's going to go first today, but I want to introduce Bang Liu, who is a program director and clinical project director at bacteriology and mycology branch at NIAID, and Donna Love, who's a mycology program director at bacteriology and mycology branch at DMID as well. The two of them are going to share most of the next hour and will just sort of provide their insights into what sort of applications they may be looking for, what tips they can maybe provide us to make these applications competitive, and really overall encourage us to seek opportunities for funding through their branch. So with that, I'm going to turn it over to Donna and Beiying. I will mute myself and turn off the camera, and then you guys can kind of alternate or go however you want to go. Donna, I see your camera's on, so I'm assuming you're going first.
Yeah, I'll go first, Peter. And thanks very much. And I appreciate the MSG and the community for making time today. We think that this is just terrific. And so what we are going to do to kick this off, I'm going to share my screen, first of all, before I forget. Let's see here. Okay, am I sharing the correct screen? Are you guys seeing the presentation?
Yes.
Yes.
Yeah. Okay. All right, thanks. So as Peter mentioned, I'm the program officer at Niad, and my portfolio is basic mycology, but we work closely with the more translational program officers, and you'll hear later on in the talk from Baoying Liu. And so what we would like to do is just introduce NIH and Niad, talk about the mycology portfolio. We would love feedback from you all since you're clinicians and deal with these invasive fungal infections. As Peter mentioned, some tips and tricks on grant applications, and then Baoying will really follow up with clinical research opportunities. And that was really, the focus of this, as Peter mentioned, going through the portfolio. It is very healthy in terms of basic research, but we really aren't seeing the number of sort of clinical and translational applications that we think maybe would be healthy for a portfolio of the size that I manage and that we manage. Okay, so just to start off a little, just to orient everybody to NIH, it consists of 27 institutes and centers shown here. You can see that in the darker blue highlighted bubbles, these are all the institutes that fund fungal or mycology research. So Niad, while we are by and large the largest institute that funds this type of research, we aren't the only institute, and that's important to keep in mind. Shown in red is the Center for Scientific Review. This is very important for folks to understand how this center operates. When you apply for funding to NIH, your application, this is the first stop for your application. And it's folks there that decide which study section is appropriate for your application, and also which institute should be funding the application. And so that's really the first stop. And then it's assigned to a study section for review and an institute for funding considerations. And so that's once those choices have been made, that's when the program officers begin to see your application. Okay. Oh, and the other important point to note here is that all of these institutes have their own budgets and their own mission statements, and the areas of research that they focus on, obviously, but they act as these little nation states. And so it really is up to you all as the investigators to sort of understand the culture of these institutes. For example, Niad, our institute is a pay line. We call it a pay line driven institute. That is, once your application goes out to study section, it's given a score. And if it's within our pay line, we pay it no matter really what the area of science, the focus is. Whereas other institutes such as NIGMS, it's a more basic institute, they really don't post pay lines. They post what they call success rates. And it gives the program officers a bit more flexibility in balancing their portfolio, and they can sort of pick and choose and actually do skips. We call them skips if they reach down and pick up an application that maybe didn't score as well as other ones because that area of research is underrepresented in their portfolio. So again, 27 institutes and centers within NIH, and they're all a little bit different. But today, of course, we're going to focus on Niad. And so for an application, usually to do well and to rise above the rest, you really, if you're applying for funding from a specific institute, you want to understand the mission and make sure that your application falls within that mission. So Niad is composed of 3 divisions, and they're all focused on leading research to understand, treat, and prevent infectious, immunologic, and allergic diseases. And so obviously Niad really does have a translational bent to its mission, but that doesn't mean we don't exclude basic biology on these pathogens. And so shown here are the 3 divisions: my division, which is the division of microbiology and infectious disease, the division of AIDS or DAIDS, and also the division of allergy, immunology, and transplantation, or DATE for short. And so I think DAIDS is self-explanatory, and you all probably know about DMID, which is my division and Bao-Ying's division. But also the division of allergy, immunology, and transplantation might have very targeted funding opportunities that might be applicable to you all. And so it's just sort of important to keep your eye on all of these divisions and sort of what they're up to. We want you all to know that it's not just me or Bao-Ying that is pushing mycology research. You have a whole team of folks within our branch that are intimately involved in this area of research. Shown here are all the different fungal diseases that we would cover and do cover. And so, as I said earlier, I'm sort of the basic mycology program officer. My portfolio is mainly the basic sciences. And we have folks that are just cover vaccines. Bao-Ying is the antifungal program officer. You also have Alec Ritchie that focuses on diagnostics, as well as our clinical team with our medical officers that review the clinical trials and clinical studies. And then down below at the bottom are 2 very important people that I want this community to know, and that is Erin Zaytouni on the left and Erica Rotterman on the right. And they are preclinical services managers, and we'll talk a little bit about that at the end. But we do provide in-kind free services to test your various therapeutic molecules, diagnostics, and vaccines. And this is a very important service that I think is maybe underappreciated in the community, but can be quite powerful to support your research. And so shown on this slide is the mycology portfolio at Niad. And the large circle shows the fungal pathogens that we support, and the wedges represent sort of the percentage of applications within the total portfolio. And so you can see that Candida and Cryptococcus really make up the lion's share of the portfolio, followed by Aspergillus, and then the dimorphs to a much lesser extent. And as I mentioned earlier, since we are P-line driven, it's really hard to balance the numbers of these applications. And I think maybe the molds have suffered a little bit because they're harder to study and there are fewer researchers. And I'm happy to talk about that later on. And then shown in the smaller circle is just the number of applications that fall within the basic portfolio, which would be mine, versus therapeutics, diagnostics, and vaccines. And just to summarize, there are 213 grants. We have 2 clinical trials ongoing, 1 clinical study, and numerous preclinical studies that have come through our preclinical services that I just talked about. And so what are the resources available to you all to support your programs? We have a number, of course, grants, the basic R01s and R21s that you've all heard of. Are really the bread and butter of Niad, but we do also have these product development contracts, and we have several that are actually on the street right now. I think the deadline is in May, but these can be fairly lucrative. The budgets are quite healthy, and they support vaccines, diagnostics, and therapeutics. We also have research service contracts. These are these preclinical services that we can do in-kind research for the community, and we'll talk about those later on. So I think Peter touched on this. We just have not seen very many research applications coming in that cover clinical research or clinical trials, and there are mechanisms available to the community. Namely, we have the investigator-initiated mechanisms, and these really form the sort of the lion's share of grants at Niad. And so I just want to spend a little bit of time about how you decode these funding opportunity announcements, or FOAs as we call them. And so shown here is just the landing page. All of the NIH FOAs look the same. And I'm showing here the PA that we have right now on the endemics. And then in the smaller square, I have the table of contents. And I know many of you are aware of this, but just navigating this, I think, can be a little overwhelming, and knowing how to get to the important parts, such as, you know, is my research responsive to this FOA? That would be, you know, in section 1. Am I eligible? We get a lot of questions about foreign applicants and foreign institutions, and that information is always shown in section 3 of every FOA at Niad, or at NIH rather. So you can always find that information there. There's always, or there could be, special review criteria if this is not an investigator initiated, or what we call a parent award. If this is an RFA, a request for funding announcement, that special review criteria would be found in section 5. And I would just encourage people, if they're thinking about applying for a particular mechanism, section 5 should be one of the first places you look. So learn how the reviewers are going to be evaluating your application before you start writing it might be quite helpful. Okay, and so again, all of the FOAs look the same. Much of the language is boilerplate, and they're all organized in this fashion. And so a big part of our job as program officers is really helping folks, you know, maybe learning how to construct these applications and getting them in a format that they will review well. And one of the most important pieces of your application is your specific aims page. This is the place where reviewers are going to go, I think, almost first. It's going to be your first impression, and you really want to spend a ton of time on this specific aims page. So obviously, you know, putting your best foot forward. You know, include the objective, or, you know, hypothesis. It seems obvious, but many times people you know, sort of skip this step, and then the reviewers are left a little befuddled. If the project is not hypothesis driven, that's fine. Just embrace it and explain why the work is important and will move the field forward. Obviously, this is where you would describe the gap that your proposal will fill. Sometimes these study sections are maybe not, they're not going to be chock full of the expertise you might be hoping for, particularly with mycology, medical mycology. You know, it's small compared to other fields, I think. And so really the challenge for you as the writer is to, you know, start out broad and then narrow your application very quickly, you know, getting to the point, but don't assume that your reviewers are going to automatically know why what you're wanting to do is important. You really do have to tell them that on the aims page. Another important point about, you know, constructing an application and designing your aims is that they need to be interrelated without being too codependent. This is one of the first things that reviewers will look at. And if they always say, if aim 1 doesn't work, then what are we left with? And so you really sort of want to design your specific aims sort of like a wagon wheel, where you have a central hypothesis and the aims are sort of like spokes on that wagon wheel, and they complement each other, but they're not codependent. And finally, I have listed here is be realistic. Do not propose too much for the time or the budget that you have requested. This is especially crucial for new investigators. Folks are quite excited. You have a lot of questions that you want to answer. And so sometimes and often you might propose a little too much. And so you really do have to narrow the scope of your application and make sure it's a really super tight super tight application. So those are just some basic tips on your specific aims page. Another thing that I always suggest with folks is, and it might sound a little silly, but if you have room, put a little graphical abstract on a picture on your aims page that sort of encapsulates your aims in one image. Reviewers have a lot of applications to get through. It's hard to keep them all straight. And so an image helps them to, hold your aims in their mind as they're reading through your application, and also helps distinguish your application from the whole stack that they have to review within a single round. And so anyways, those are just some tips to help you sort of create a stronger application. And then I just wanted to spend a little bit on frequent reviewer comments. What are some common obstacles encountered in study section? And without a doubt, these are sort of the top 5 comments that particularly young investigators will struggle with. That it's too ambitious. Again, the scope has to be a little bit more narrow than you might be comfortable with, and quite tight. Prioritization. That's sort of low-hanging fruit for reviewers. If you are proposing an area of research where you will be collecting a lot of data, if you're doing any kind of omics, if you don't have a prioritization scheme, it's sort of a fatal flaw for your application. They'll call it a fishing expedition. And then alternative approaches. I think the reviewers really want to see that you are have thought through all the experiments. If something doesn't work, what are you going to fall back on? And then, not responsive if it's not an investigator-initiated mechanism, but something that is one of these RFAs. You want to make sure that you're being responsive to what's in the RFA. So again, reading through the whole FOA very carefully is crucial. So I think the review process can be sort of a little mysterious for folks, and NIH, I think in particular, we have many iterations of how we asked the reviewers to critique the application. And so I just want to go over those 5 review criteria, and then how the overall impact score is sort of generated from those 5 review criteria. So the assessment of the likelihood for the project to exert a sustained and powerful influence on the research fields involved. That's what the reviewers are asked to consider when they're giving an overall impact score. So the overall impact score is not mathematically related to the individual criterion scores that the reviewers will give you. This overall impact is just, will this move the field forward? And so the likelihood that it will is primarily derived from the investigator, the approach, and the environment, these criteria. And really the approach is going to be the one that will be score driving. And then, will this application have a sustained and powerful influence is primarily derived from the significance and innovation criteria. And so these are the 5 criteria that they will go, and you will get, tons of information under each of these criterion. However, significance and approach are really the main drivers. Sometimes reviewers will get a little distracted because maybe for innovation, they gave you a 4, and an application doesn't really have to be innovative to, have a high overall impact score. So it's important to focus on significance. Did they give you a poor score for significance? If they did, then that is sort of, a warning flag. You need to really address that. Perhaps you didn't give them enough background, so they didn't understand the importance. And then again, the approach. The reviewers do want you to succeed. They want the science to improve, and so you really do need to listen to what the reviewers are telling you. And it's our job as program officers to sort of help you synthesize this information and to improve your application. So shown here is our fiscal year, the NIH fiscal year. And I put this up because I think it's really important for folks to understand sort of the rhythm of our fiscal year as you're putting in these applications. And shown all the way over on the left, we have the review cycle. So we have 3 cycles per year. Those also are correlated to our Nyad councils, we call them. So your application, once you submit it, it goes to a study section, and that is reviewed there. That's called our first level of review. And then the second level of review is council, where we have another panel of esteemed scientists that sort of evaluate these applications en bloc and approve them. And so we're coming up actually on our June council, shown at the bottom. And so what's important to know about this? If you submit for cycle 1, these are the winter due dates. It's reviewed in the summer. And then look at the possible payment. It can be from October, which is the beginning of our fiscal year, all the way until September sort of the following year. So the maximum time from submission to possible payout is 1.5 years. So this is really critically important for folks to understand this, especially as a new investigator, or when you're trying to time your funding for your laboratory. And then the minimum time is cycle 3. The due dates are in the fall, and you have possible payout, the following summer. And so that's the shortest cycle. For me, I always tell folks, if it works out, it's really great if you can submit in cycle 1, it's reviewed, you get your summary statement back, and then you can come in for the 3rd cycle. And so you have 2 chances within the fiscal year, which is often needed, and that's just sort of very convenient. Okay, so I just have a few more slides to go over. Communicating with program officers, do we welcome communication from applicants? And absolutely, I think it's very important. We can help you determine if your research fits the mission of our institute. Often, and maybe not with this crowd, but more basic researchers, their applications are somewhere within NIGMS, general medicine, and our institute. And it's never good to have an application that's out in no man's land. And so we can help you sort of with the grantsmanship to kind of, pitch your application to the mission of the institute. We can help you find possible funding opportunities that might be more appropriate. Our biggest job, I think, is really discussing the review and helping you decode the summary statement and what's in that. And then finally, inquiring about end of year funds. And so I have this little quote down here, it ain't over till it's over. And for, in the previous slide, I just showed the fiscal year, that can be 1.5 years. So it's always good after you get a score to say, am I close to the pay line? Could I be considered for end of year funds? Those are conversations that you should be having with your program officer. And so here, this slide, communicating again with your program officer, how should applicants prepare before speaking with their program officer? And I really think that shown here on the right is sort of the 7 levels, I think, of grief. And sometimes I think that applies to when you get your summary statement back. Folks, it's sometimes can be quite shocking, the comments from the reviewers, and you're super angry. And then maybe a little depressed that they didn't, like your science, or they didn't find it significant. However, it's always important to transition all the way through. And when you're feeling a bit better, it's time to reach out to your program officer and to talk about that application, because you can always improve these. But you do need to be open to constructive criticism. Some mistakes that I see with folks is that instead of putting in a resubmission, they just keep putting in a new application if they're not discussed in review. Only 50% of applications are discussed in review. And so if you find yourself not discussed, there's still tons of good information there in your summary statement and ways to improve. And the reviewers do reward you resubmitting your application and coming in a second time, and we can help you with that. I've seen many applications go from not discussed in the first cycle to making pay line in the second cycle. And so it's really, you need to think of it as an iterative process, and the applications do improve going through this process. So these are just some examples of current FOAs that are available, and I just show them here. But again, Bao-Ying and I, what we want to do with our time here is just to introduce ourselves and to have you reach out to us and set up a phone conversation. We'd be happy to talk about your science and then guide you towards opportunities that would be appropriate for your science. And other ways to find out about funding opportunities are shown here. We would encourage everybody to sign up for the Nyad funding news. They're on Twitter. They will send it by email, I think even maybe Instagram, just so you're aware of what's coming up. The other really important sort of tip to be aware of is the Nyad Council cleared concepts. So as I mentioned earlier, we have council 3 times a year to go over all the applications that have been reviewed. And that's also the time when we talk about concepts that are likely going to be funding opportunities. And so if you read the summary from all of our concepts, you'll know what's going to be published as funding opportunities probably a couple months before they actually hit the street. So that just gives you an additional time to get ready. And again, our division, DMID, DAIDS, and DATE, have their concepts posted. And some of those are really big, $1.2 million budgets that might be applicable to this community. So again, I would encourage everyone to take a look at the council concepts when they do come out. And then finally, I know you all know about NIH Reporter. I just show a little snapshot from the new and improved Reporter. At the bottom of the slide, it's I think a little bit improved. It's a little more graphical. You can find out what other folks are working on in your field. You can look at their budgets and see how their budget requests, that kind of thing. And I think there's also a matchmaker tool here where you can find program officers across NIH. And so I think it is a really powerful search engine that you probably want to familiarize yourself with. Okay, I just want to spend a few minutes going over our preclinical services before I turn it over to Bao-Ying. And again, here are the 2 different websites. I think that that's kind of hard to navigate, so that's why I show them here. If you look under Niad, nih.gov, research resources, and then therapeutic development services are the 2 ways to access what we have available on our website. Again, this is what it looks like. You can put in, your area and pull up any sort of additional resources that we might have available. And these, again, are sort of in-kind free resources. And so again, like one of the resources that we have available to the community is really a whole suite of bioinformatic tools and resources. For example, we do crystal structures for free for folks if they have a protein that they can express fairly easily. We have contractors that will provide crystal structures for you. We also do support for therapeutics and diagnostics, folks that are developing monoclonal antibodies or recombinant proteins, siRNA for therapeutics. We also have supports for those. For example, we do MIC testing for virtually anyone. We have a whole battery of fungal pathogens that we will test and see if your molecule is effective. We also do tox testing. We have animal models, and those different animal models are listed here. We PK, PD, even all the way up to phase 1 clinical trials for therapeutics. And so again, I just want to stress we have a whole sort of team that manages these. They are free. And you don't have to be, I think this is on the next slide. Oh, I just want to show you here, these are all of the new therapeutics coming down the pipeline for fungal diseases. And I'll just say that many of them have come through our preclinical testing. So this is a really, I think, very useful tool for the community. And the eligibility criteria, it's anyone, basically. You don't need to be funded by NIH. It can be, you can be an international institution. These are meant to be gap filling. We can't do everything, but you know, we can do help you with some areas. And I just want to say in closing that these are reviewed in-house. It's usually, it takes maybe 6 weeks to find out if we can, you know, do some of this work for you. And so I think it's a nice tool for the research community, and it's not quite as long and drawn out as the regular grant process. And so with that, I think I will hand it over to Baoying to talk about more of the supports for clinical research.
Thank you for the opportunity to present. Again, I'm Baoying Liu. I'm the Antifungal Therapeutics Program Officer of Niad. In today's talk, I'm going to focus on our funding opportunities on clinical research. Next slide, please. First, I would like to introduce the investigator-initiated clinical trial, IICT. IICT is a mechanism for investigators to apply your own clinical trials outside Niad's networks. Under IICT umbrella, there are 5 funding opportunities for you to consider. The first one is the the standard R1 research project grant. So the current FOA on street is PA 2183. So if you Google Niad with this PA number, then you will find the current funding opportunities. Applications that respond to this FOA is usually reviewed through CSR study sections. This mechanism is best for clinical trials that are not high risk. So in the next slide, I'm going to tell you Niad's definition of a high-risk clinical trial. So if your budget request is equal or over $500,000 direct cost in any project year, then Niad's pre-approval and prior consultation is required. So you need to do that at least 10 weeks before the submission due date. The pre-approval is just to make sure that Niad agrees to accept this assignment. It's not a scientific or technical review. Therefore, it cannot replace the peer review process. Even your budget request is less than $500,000 direct cost per year. So the prior consultation is also very strongly encouraged. The purpose of the prior consultation is mainly fourfold. First, so we want to make sure your proposed clinical trial address our mission. So you and the program officer can determine the risk level of your clinical trial. And then we will identify the best mechanism for this proposed clinical trial. And additionally, you and the program officer can review the information and documents that will be needed in your application package. So the R1 mechanism can support up to 5 years. However, if your project period is longer than 5 years, then we have the second mechanism we call Niad extended clinical trial. It's also R1 mechanism. Is best for project period of 6 or 7 years. And of course, we also have R21 mechanism to support small project proof of concept projects. The last 2 mechanisms here are cooperative agreement. So I'm going to provide additional information in later slides. Next slide, please. Donna, next slide, please.
It doesn't, now it's stuck. Wait, let me see. Okay.
Perfect, thank you. So this is Niad's definition of a high-risk clinical trial. A high-risk clinical trial should have 1 or more of the following attributes. If the clinical trials involves a non-routine intervention, That is an intervention that would not otherwise be provided for the disease condition under this study in the same facility. Or your proposed clinical trial involves administration of a licensed product or administration of a licensed product for an unapproved indication. The above three criteria determines whether a clinical trial is high risk or not. Next slide, please. So here I want to provide additional information on one of our clinical trial implementation cooperative agreement. We call it UO1. UO1 is the best mechanism to support high-risk clinical trials. It's equivalent to the R1 I just mentioned. The maximum period is also 5 years. The major difference between R01 and the UO1 is that UO1 has substantial involvement from Niad. That means that Niad staff will participate, coordinate, assist, and guide your research activity. To give an example, Niad may have access to the data you collected from the clinical trial. Our medical officer may be involved in the clinical monitoring process. We may help you to identify resources that may be needed for your clinical trials. And there will be a regular meeting between the study team and the Niad to review the project status. Again, if the budget request is over $500,000, equal or over $500,000 direct cost in any project year, then the prior consultation is required. You need to do that at least 10 weeks before the submission due date. I also want to point out a couple of items here. First, all clinical trial planning activities must be completed prior to the time of application submission, and the investigators must be ready to implement the proposed trial at the time of award. The expectation is you need to have the IRB approval prior to the award. The second point I want to mention is inclusion, a clear and well-defined milestone is the key. A milestone is defined as a scheduled event in the project timeline, signifying the completion of a major project stage or activity. For example, you need to define the 25 or 50 or 75, 100 patient enrollment timeline. And you better, in your package, you better build in a contingency plan and could have a discussion of alternative approach. And finally, the foreign institutions are eligible to apply for UO1. Next slide, please. So in this slide, I'm trying to provide some information on our small business clinical trial implementation cooperative agreement. We call it U44. U44 is a very attractive mechanism for the small business entity to conduct your clinical trials. Fungal diagnosis, therapeutic, vaccine, they are all Niad approved waiver topics. So you can request up to $1 million per year. That's $1 million total cost for up to 3 years. In terms of application type, there's new phase 2 or phase 2B. So depend on whether you have prior SBIR grant on the same topic. So it's a little bit confusing, but please reach out to us so we can help you to interpret and lead you through the application process. Again, the U44 support hypothesis-driven, milestone-driven clinical trials. Clinical trials that are not high risk are accepted through U44. But this program encourages high risk clinical studies. Again, the prior consultation 10 weeks before the submission due date are strongly encouraged. Next slide, please. In addition to the above 5 implementation grant, Niad also has clinical trial planning grant. We call it R34. So you can request up to $150,000 direct cost, and the maximum period is 1 year. So one thing I want to mention is that R34 is not a pre-requirement for the ICT implementation awards. And at the same time, funding of 34 does not guarantee or imply funding of a subsequent year-one. So of course, R34 supports timely development of all materials required for implementation of a future clinical trial. For example, you can use this 1 year to build a team, to develop your clinical protocol, develop the informed consent data collection form, and some such. And foreign institutions are eligible to apply. Next slide, please. So in addition to the grant mechanism, Niad also have contract mechanism to support clinical trials. And this slide is an example. It's about phase 1 clinical trial units. This contract mechanism support the design, development, implementation, and the conduct of phase 1 clinical trials. It support safety and PK/PD study in healthy volunteers only. In this slide, I also listed 3 locations that conduct phase 1 clinical trials here. One thing I want to point out is for clinical trial supported through this mechanism, DMID will sponsor the IND application for the study. DMID will provide safety oversight, clinical monitoring, data management, and the regulatory management. The last date for the current contract is December 2021, the end of this year. Next slide, please. So after the phase 1 unit expires, then there will be new contract mechanism called early phase clinical trial units continue. These early phase clinical trial units will support the implementation of phase 0 to phase 2 clinical trials in both healthy and disease population. So it has an expanded scope. And then this contract can also include bioanalysis. This clinical trial unit support both licensed and investigational therapeutics, vaccines, diagnosis, and adjuvants. The current status for the early phase clinical trial units is currently in the solicitation stage through the request for proposal, RFP. The planned contract award date is January 2022. Next slide, please. And then lastly, I also want to mention these 2 mechanisms for Niad to support clinical trials. One is consortium, and the other one is leadership group. So the consortium, we call it IDCRC, Infectious Disease Clinical Research Consortium. This clinical trial network was established by Niad in December 2019. It includes the previous VTU sites and also a new leadership group. There are 6 expert working groups within IDCRC, and one working group that particularly relevant to our fungal community is the Emerging Infectious Disease Working Group. The pathogens and diseases include coccidomycosis and Candida, especially for Candida auris. And here I listed the website, so you can have a clear picture in terms of IDCRC's vision, structure, and application process. And lastly, can you go back? Lastly, I want to mention, Donna, if you can go back 2 slides. Yes, so mention the ARLG, the antibacterial resistant leadership group. So ARLG is created in 2013, mainly to address antibacterial resistance. So it do the clinical trial, clinical study on antibacterial resistance. In his new version, because Candida auris has attracted a great deal of public attention. So proposals addressing Candida auris may be considered by ARLG. ARLG does not have a fungal specific subcommittee, so fungal related projects may be evaluated on a case by case basis. So I also show the website here. Next slide, please. So these are the resources that are available for our community. I hope you can consider to apply. Please don't hesitate to email Donna and myself. So we will be very happy to lead you through the application process. With that, I conclude our presentation. Thank you very much. The picture is our bacteriology and mycology branch picture. Thank you.
Well, thank you, Donna and Beiying. That was really insightful, very helpful, very well organized, and I think you covered a lot of different territory. So I want to thank you for putting the effort into that. We're in the question and answer session. So if you have questions, use the chat or the raise your hand application, and then we will address those. Deb, are you seeing anything?
I am not seeing anything. The raise your hand function is along the bottom of your toolbar under the reactions tab. So
Okay.
if you have a question, if you want to raise your hand, we can unmute you and you
Okay.
can ask your question
Here's
live. I do see something in the chat, Pete.
Yep, I do. Okay. So Erica has a question. Could you please define the boundary between basic and translational research? I think a lot of us struggle with that. I see Donna laughing. Apparently you struggle with it as well.
Right. Yeah. I mean, I think that that's a little tough to define. I think that it's not so important to define it for us. I guess I mentioned it early on in my talk, but Niad covers, basic science in the context of infectious disease. So for example, fungal biology, I think basic physiology, those kinds of things. As long as it's related to an infectious disease, it would be covered within Niad. And when I was talking about, I cover basic biology, and then there are folks in our group that cover the more translational science, then we break it up by, is it a therapeutic, is it a vaccine, that kind of thing. So I hope that answers your question. I don't know, Beiying, if you have anything to add to that.
Yeah, I agree. Actually, it doesn't matter from investigator point of view. So it matters when Nyad make assignment. For example, if it's therapeutics, it will go to one program officer. If it's diagnosis, it will go to another. But we do accept all fungal related research proposals.
And it does always help to have an eye towards the translational applications. You don't want to oversell your proposal if it's basic fungal biology and you say, oh, I'm going to identify a molecule. But if it makes sense to do so, then programmatically, those kinds of things can help us sort of champion your application. So it does, there's always a grantsmanship issue, I guess, in just selling your application.
Somebody wondered about slides. These will be posted on the website, the MSGERC.org website. Tom Patterson has a question, and it relates to timelines. It's a really good one because I guess for the longest time we thought that maybe the interval could not be compressed, but we saw that during COVID it actually was compressed substantially. And so, what can we do about this? That the timelines have been an issue, and anyway, is there discussion internally about shortening these timelines?
I don't think so. I would tell you that the timelines were compressed for the COVID applications, and it came at great cost, I think. Not only in terms of maybe review quality, but also just administratively, it was very difficult. So I don't think that there are any discussions to compress the timeline. I know it can be quite long for folks, but I don't think that there is, but that might be a little bit above our pay grade.
Okay. All right. What's your experience with the, I'm not familiar with this, but I'm sure you are. What's your experience with the Stephen Katz Early Stage Investigator Research Project Grant? Any thoughts about that?
I don't know yet. I think it's too new to know how the reviewers will react. We have a few of these type of mechanisms where they're not asking for a lot of preliminary data, that kind of thing. And so I think it's going to take several cycles before the reviewers really know how to review that. And I think the applicants know how to write the applications. As you all know, R21s don't need, they don't require preliminary data, but they always do better with preliminary data. So I think that one is still too new to offer any useful comments.
And what about the question, how do young investigators or early investigators get involved or get to know the IDCRC? And I know they have a structure, they have sort of a board, if you will, and then they have sites. And not all board members are from those sites. So how does a young person get plugged into IDCRC?
That's a great question. Bao-Ying, do you want to take a stab at that one?
Yeah, that's a great question in terms of IDCRC. So if you go to their website, you do see their structure. They have 6 working group. They also have a leadership group. So in terms of young investigators' involvement, I think, like I mentioned, it's only established in late 2019. So I think both DMID and also external leadership group, they are still exploring how to make it more accessible to the investigators. So I believe they do have some training program. So just go to the website and see what kind of opportunity they have. So we do also have program officers who is responsible for IDCRC. So if needed, I can connect you with program officers related.
And I would encourage people to be bold. You know, as Bao-Ying said, go to the website, find the names, and just try to initiate contact with those folks. I think that's reaching out is a really good first step.
We have time for a final question, and then we'll take a 10-minute break. Which study section would be best for targeting host proteins, small molecules / antibodies for the treatment of fungal infections?
Bao-Ying, it looks like this is you.
Yeah, so study section. So I believe for the therapeutics, usually application will go to 2 major study sections. In the past, a lot of application will go to DDR, which is drug development and resistance study section. But recently, our Center of Scientific Review, they have the reorganization. So it's not official, but the new director has a vision that for fungal related applications, it will be reviewed through a new study session called AIDC. So it's not official yet, but I think it's this, they are exploring the review process through this round. So basically, all fungal or eukaryotic application will go to one study session to be reviewed. It's called AIDC.
Once again, I'm really pleased with the presentation, Donna and Bang, and I think this is very helpful. It's a great start to this program today. And so at this point, let's take a 10-minute break. We will start promptly at 10 minutes after 1, and we'll get started on the second hour of this program. Thank you very much. So I would ask everybody to turn off their cameras and mute at this point.
Welcome back, everyone. I have 2:10. Dr. Pappas, would you like to get us started again?
Yes. Yes, I would be pleased to get us started. So this is section 2 of our session today, and this is the advantages. This we'll just call this section the advantages of NIAID funding. We've selected 4 speakers, 2 intramural speakers, Mihalis Leonikis and Stephen Holland, and then 2 extramural funded investigators, David Andes and David Bulwer. They'll each take 10 minutes, and then we will have a session for questions and answers at the end of all 4 of these presentations. So with that, Mihail Leonikis, thank you so much for being with us today.
Thanks, Pete. What we've talked among ourselves and decided to do is maybe give a very brief overview, 30,000-foot overview of what we're doing intramurally. As you'll see, there are already collaborations with members of MSG, and this we thought could serve as an opportunity to discuss about other areas where we could converge. So I'm an ID trained physician and immunologist, and I came to NIH in 2007. So this is the building 10, which is a clinical center of about 300 beds, the largest research hospital in the world. And our labs here. So I came here as a clinical fellow in 2007. I did research and then stayed about 8 years ago as a tenure track and now tenured. And now, they don't seem to find a way to get rid of me. So I've been very happy in here. And what I'll try to do is to give you a little bit of an overview of how we approach host defense against common fungal pathogens. These are the 2 that we mostly focus on, but not exclusively. And we do that in a way that takes advantage of this clinical center and the resources we have here. So enrolling patients who have rare but very enlightening genetic disorders that lead to fungal disease. Now, many of those are actually phenocopied by drugs, inhibitors that we give as physicians to patients who suffer from different diseases. And at the same time, we go to mice and try to interrogate mechanisms and going back and forth, try to understand how we all are mostly resistant to fungal disease, but yet infections happen when host immunity is perturbed. So very, very briefly, again, over the next 7-8 minutes, I'll just give you a very high-end overview of some of the things we've been working on, and predominantly on Candida, with just a small slide at the end on Aspergillus. So Candida comes in 2 flavors, as you all know, and those depend on very different things for host defense. So phagocytes are important for systemic, and lymphocytes for mucosal disease. So in the systemic arena, we've really try to understand fundamentally how these gray neutrophils that you see here would attack in vivo these red Candida. We all know that neutrophils are absolutely important in humans because when patients lack those, they get severe fungal infections. And we've spent a lot of time over the last decade in a summary slide that is not important to go through the details to identify how these phagocytes act. Importantly, through, again, some of the collaborations with John Perfect in this particular case, we've been able to uncover at the population genetic level how some of these might confer susceptibility to fungal disease. And importantly, as I mentioned earlier, through the advent of several of these biologics, some of these are actually likely to represent novel iatrogenic risk factors. This is highlighted in this slide where mice that lack complement receptor all die from fungal disease. Actually, humans in the ICU who do not mount proper C5 levels at the onset of infection, again, this is in collaboration with John, they don't do very well. Now, that has led us to explore some other pathways that have both an inherited and an iatrogenic flavor to them. So this is the C-type lectin receptor pathway. Many of you who study immunity do study this. SIC is an inhibitor targeted molecule that, again, is amply used in several studies. And CAR9 is a disease that we've come to study some of these very, very impressive patients. So here are a couple of examples of a girl at the time had this enormous fourth ventricle due to this brain thrush. This is brain here, third ventricle full of fungi. Sorry for these images, but this disease can also cause severe skin infection. This is a nail fungal trichophyton. This is a cucumber fungus that shouldn't do this to any human, and yet in this disease it does. And as we've enrolled over 150 patients, we've come to realize that most of them actually don't have mutations in what you would expect, indicating that there are many more things that can be discovered, and when they are discovered, to figure out how. At the same time, we focused on mucosal disease, where IL-17 has been shown in both mice and humans to be absolutely critical. However, what we've done is taken an approach, again, through some of these very striking human inborn errors of immunity to phenocopy them in mice, and to realize that some of these disorders are 17 deficiencies, but some of them are not. And this is a very brief highlight of a recent study we published showing that in the setting of air deficiency, the significant susceptibility of these mice to Candida was actually not because of IL-17 deficiency. Rather, it was a setting by which excess interferon gamma by T cells was driving the disease. So we're all familiar with T cells being protective against mucosal disease. In this particular case of immune dysregulation, they're actually doing harm. And then doing this through production of interferon gamma, such as if you eliminate interferon gamma, genetically or pharmacologically, you restore protection, leading us to produce this graph that says you can get in trouble with mucosal disease by lacking IL-17, but also in certain settings by having too much of inflammation. Now, in the setting of COVID, I think we're all becoming more familiar to how you can actually get infection and bad outcomes, not because you're lacking an immune factor, but rather because of excess inflammation. I think this is one of those examples. And it's likely to have other settings that follow that, whether that's graft versus host disease, or even in the case of HIV, where the same pathway in humans appears to be turned on. And because Th17 deficiency alone is unlikely to drive mucosal candidiasis in humans, because patients who lack CD4 T cells, they have ICL, they don't typically get Candida. We've now started looking at this in the macaque model of oral thrush. Last slide I have for you is molds, which are also a major focus of our lab, where again, through human and mouse studies, we've been able to identify new genetic underpinnings. Interestingly, in these three cases, extrapulmonary aspergillus, we're all familiar with this being inhaled molds and causing predominantly lung disease, but in certain settings that can be extrapulmonary. We mentioned earlier about major iatrogenic risk factors. I think you're all familiar now about ibrutinib and aspergillosis and how actually BTK is very important in neutrophils in an unrecognized role of this molecule in these cells. And I mentioned earlier about eculizumab and Candida, but there's also a susceptibility to aspergillus. Actually, the FDA changed their package inserts to reflect that. And we've recently expanded to work with pheohyphomycosis, and some of these were Sanjay and Pete trying to get through the mycosis study group more patients who, again, out of the blue, would develop these nasty infections. So our overall goal has been through identifying molecules in mice to see how we could potentially translate that to humans, either in the setting of re-stratification or therapeutics, understand better the surge of these iatrogenic risk factors as we are in the era of biologics, and I think a new subspecialty of infectious disease, quite frankly. When we study these rare patients and we see these patients here at the clinical center, we try to understand pathogenesis, but also try to be physicians that try to understand the disease in order to develop targeted therapies, and sometimes we're lucky to be able to do that. So with that, I would like to show you the most important slide. We're very collaborative, not only in my group, I think overall. You can see here a lot of your names on the extramural arm. We're open to ideas and ways that we could interact in our studies. And I think with that, I will pass the torch to Steve, and I'm exactly on time.
Thanks, Mihalis. So my name is Steve Holland, and I'm an investigator here in NIAID and currently the scientific director. You know, I came here in the last century. You can relax. I'm not going to show you any slides. The more I thought about what slides to show you, I knew I'd just get confused trying to talk to you about data. But I don't think that's what I'm really going to be able to help with here. I'd love to tell you about the research we're doing. I think it's really exciting. But I want to just remind you about a couple of things. I want to take off from where Mihalis left off and remind you that we are here as a national referral hospital, a hospital that is, yes, the largest research hospital in the world, but it's a hospital that doesn't have an emergency room. And that means that we are entirely dependent on you, our colleagues in the communities, our academic communities, our regional communities, but in really the community as a whole for our interaction and to be able to do the kind of work that we want to do. And we charge nothing for having patients come here. We are desperately interested in collaboration with you and with others, both in this country and around the world. There is no charge for patients. We can often help to bring patients in by paying for their travel, depending on a few parameters. And we really don't care whether they have insurance, and we also don't care about their citizenship. That is, whether they're citizens of this country or another country, as long as they can travel here and get here and be in this country, be legally able to come on campus. That is no outstanding warrant, and I don't say that facetiously. That's really all that matters. And we are really keen to continue these collaborations. So I'll just say that, you know, I came into this all accidentally. I started out as an infectious disease person who trained in HIV and wound up working on primary immunodeficiencies, predominantly chronic granulomatous disease. Through a mistaken identification of a colleague, I wound up working on mycobacterial disease, that it was mistaken it was chronic granulomatous disease. It wasn't, it was something else entirely. And then through an interest in mycobacterial disease, wound up interested in cocci and the extraordinary things to learn about coccidioidomycosis are really at the heart of where we want to be, I think, as an institute, understanding host factors, understanding how we take those diseases that have extreme phenotypes, like disseminated cocci, and then by nibbling in from the edge, sort of, you know, coming from the edge of the cookie toward the center, trying to figure out what are the core features that are important for cocci in the general populace. And cocci is a great example because it's got regional components, it's got gender components, it's got ethnic components, and we now know it's got really important genetic components that underlie susceptibility, very much like the factors that Mihalis pointed out to you in Candida and some of the dermatophytes. There are some really interesting genetics behind this. And our goal is to take those genetics from the rare back to the common. I'm sure that's your goal as well. We can help because we can sometimes set up collaborative opportunities, UO1 grants, and collaborative activities in R01s to really begin to partner with you to be able to work on these things more forcefully. So I think I'll stop there, except to say that you've got Mihalis doing the work he's talked about on Aspergillus and Candida. My own group is working on Cocci and a little bit on Histo. Peter Williamson's group is working on Cryptococcus. We're very keen on trying to do this. And then we've got a major program in transplantation for the immunodeficiencies, especially those immunodeficiencies in which these infections are so rife. So our goal is to be a national and an international resource, and we'd love for you to take more advantage of us. I'll stop there. Thanks.
So Pete, I think I'm up next. If I can
Yep, I think that's right.
share my screen here. Are you guys seeing?
Yeah, we can see it.
The presenter mode? Okay,
Got it.
It's full screen.
thank you. Okay, so my name's David Andes. I'm at the University of Wisconsin. I'm an infectious disease clinician with also, I consider it a translational research laboratory. And so my interactions with Niad have been twofold. One as an investigator, and second as a reviewer on a fungal study section, primarily the study section that reviews the basic science and translational studies. And I'll reiterate what Donna said earlier. I mean, my experience from my interactions with Niad have been really awesome. I think from the funding standpoint, they're interested in clinically relevant questions. I think they're interested in translational approaches, team science. They're willing to fund ideas that most others won't. And that frequent and early, especially interaction with program has been super helpful. It's been very transparent and honest. I think people don't take advantage of the other in-kind resources they have available. And then lastly, through interactions like this, it's identified a lot of collaborative connections for our group. And so like Mihalis and Steve, I think what I thought I would do is talk about a little bit of our interest in two of our programs that I think illustrates sort of my, these experiences. So we're broadly interested in mycology, but most of our Niad funded work has been with different Candida species. And within, the fungal, the Candida as a pathogen, we're primarily interested in therapeutics and specifically why therapeutics fail, with a long-term goal of identifying either drug targets or new therapeutics to overcome resistance. most of my questions are clinically or translationally oriented, informed, frankly, largely through interactions with and studies from MSG. But most of my funded research has been at the bench or in animal models. And we're hopeful and looking for collaborations to now bring some of the promising leads we have back to patients. And so hopeful that can involve the mycosis study group and some of Bao-Ying's resources. So within Candida, the primary disease state that we think about in one of our programs is the biofilm disease state. Our interest formed in large part from MSG studies that have demonstrated the importance of this Candida lifestyle, illustrated in this data set from MSG on management of these biofilm infections on central venous catheters, where drugs alone don't seem to sufficiently clear these infections. And again, most of our questions around this biofilm state are, why do these antifungals not work well to cure these really recalcitrant infections on predominantly medical devices? And so our first actual funded project from Niad was to try and understand the mechanisms underlying resistance to antifungals when these species are growing as biofilm. And what we found in large part is that resistance to antifungals is linked to a unique feature of the biofilm lifestyle, and that is a protective material that the cells secrete and encase this community in, as illustrated here. When we expose intact biofilms to an antifungal, really all of it gets sequestered or like a sponge into this encasing extracellular matrix. So we spent a large amount of our first funded project trying to identify the components of this extracellular matrix that seems to protect the community from antifungals, and found that it's pretty complex, but we did find one unique polysaccharide complex that seemed linked to this protective feature of the extracellular matrix, and have spent a lot of the last 5 years trying to identify the pathways important for production and assembly of this extracellular matrix. And we've identified a number of genetic pathway components, more recently finding most of that linked to delivery of these extracellular matrix components by the extracellular vesicle pathway, and are currently attempting to identify therapeutics to interrupt this pathway, with hopes of identifying antifungals that can either prevent or effectively treat Candida when it's growing in biofilms. Sorry, it looks like the Microsoft is not liking me today. Y'all see that again?
Yes.
Okay, sorry about that. So our second project was to try and take these drug targets and identify novel antifungals, again, targeting things like the biofilm state. And the second project is philosophically centered on this process called symbiosis. And I think this is an example of Niad being willing to fund projects that other groups wouldn't. And so again, this is based upon the discovery of one of my colleagues that leafcutter ants, so he's a evolutionary biologist, Cameron Curry, have evolved with this material, which is actually a bacteria. And what he found is that the ants have evolved with this bacteria in part because this bacteria produces natural product antifungals that kill fungal pathogens in the environment, including the crop that these ants farm and use for food. And so we found that this symbiotic environment where we find novel anti-infectives from bacterial symbionts isn't unique to ants. In fact, it's found in most insect species and other animal species as well. And more recently, we've found a symbiotic microbiome component of a sea squirt that makes an antifungal that we're quite excited about and hope to, again, be able to bring, pursue studies to bring this back to the bedside soon. So this antifungal is called Turbine Mycin, and it's named after the sea squirt that this component of the microbiome resides in. And this bacteria called Micromonospora produces this chemical, which is Turbine Mycin, that we find active against a wide spectrum of drug-resistant fungi, including Candida auris. And in preliminary PK/PD studies, we find that really low doses of Turbine Mycin effectively eradicate or reduce the organism burden in immunocompromised mice models at pretty low doses. And we found that we can give 1,000-fold greater doses in the mice pretty safely, which is always a big hurdle with antifungals, is hurting the fungi without hurting us. Interestingly and sort of serendipitously, our mode of action studies suggest pretty strongly that this antifungal works by inhibiting the extracellular vesicle trafficking pathway, illustrated in this sort of cell biology study where we have GFP labeled SNCC1, which usually resides in the ER and Golgi, and during growth of the fungus is transported to the plasma membrane, particularly in the buds. So you can see in this control, a lot of the green goes to the plasma membrane in the buds, but with exposure to low concentrations of Turbine Mycin, it really all stays in the ER in Golgi. And so I say this was serendipitous because it seemed to intersect with our bio work, because we had just recently found that a lot of the delivery of the protective matrix occurs through vesicle trafficking. So we've recently studied the effect of Turbine Mycin on biofilms and found that, as we would have expected from our study in planktonic cells, that when you expose biofilms to Turbine Mycin, you reduce the production or delivery of extracellular vesicles outside of the biofilm, which in turn eliminates the delivery of the extracellular matrix. And then, as you would expect, with increasing concentrations, you find that Turbine Mycin can do its job and kill the fungus, even when it's growing in a biofilm state, whether that be a 96-well polystyrene plate or in a vascular catheter model in rats where exposures to low doses of Turbine Mycin really clears the biofilm from the intraluminal surface as opposed to control animals. So we're at the point now where we're hoping we can, again, move from the bench closer to the bedside. So we're at the point where we're hoping for clinical and translational collaborations, both with Niad through discussions with Donna and Baoying, as well as folks in the mycosis study group. And so I can envision a number of clinical collaborations that, you know, like the opportunities that Mihalis and Steve mentioned, give our laboratory and others access to clinical samples, so we can hopefully find a clinical corroboration of our mechanistic studies, and perhaps more importantly, allow us to develop or pursue additional clinical development of this antifungal for a variety of systemic fungal infections, but certainly including biofilm treatment studies. And so, you know, I think to me, you know, these sorts of projects formed, you know, sort of my opinion of Niad. They've just been super supportive. I think these illustrate that they're interested in clinically relevant questions and approaches that are translational. I think a lot of this getting funded was the fact that we were looking for solutions, therapeutic solutions, that we utilize translational approaches like animal models that mimic disease in patients. And I think they really, really like team science. And so I think MSG collaboration with those at the bench, I think has a lot of opportunities for resources to move studies forward. And again, early and frequent communication with Donna, Baoying, and others. I just can't say enough about how important that has been in forming ideas and the application process itself, and in identifying, again, collaborators to move this forward. And so I think with that, I'll end and thank the team of folks that have helped with the studies I've presented, and thanks both to Pete and the MSG and Donna and Baoying and Niad for all of their support. I like that.
Great. So as I'm David Bulwer, I want to sort of take off where David and Andy's spoke, and that our team really focuses on sort of the clinical and translational research in Cryptococcus, and sort of more on the clinical side. And so you heard a little bit about the preclinical aspects, and just a little bit of background that I'm sort of going to talk a little bit about what we do, but also sort of perspectives for new investigators as to some pearls for NIH grant applications and sort of my career and how that's progressed with funding opportunities. And so just for background, I graduated from med school in 2000. I did a med-peds residency followed by a fellowship in Minnesota. And I love the weather in Minnesota. And so I stayed on as faculty in 2007 and was promoted. And I'm still here along the way. And so what I'm probably best known for is actually my COVID work with hydroxychloroquine, but that's not actually my passion. So my passion actually has been about meningitis and really about Cryptococcus. And so why Cryptococcus in particular? So it's just something that, you know, as a U.S. trainee, I saw a couple cases of Cryptococcus, but it, you know, wasn't a major issue. And then I went to Africa and I realized, wow, this is a major opportunistic infection. And so when you look at the etiologies of meningitis in Africa, Cryptococcus is the most common cause of adult meningitis. And in Uganda, where we work, it's more common than all other causes of meningitis combined. And so there's a large burden of meningitis. And so for those that aren't aware of where Uganda is, it's in East Africa. And along the years, I've collaborated with a number of other colleagues in South Africa and Tanzania and Ethiopia, but most of our bread and butter has been in Uganda as far as most of our clinical trials and our pathogenesis work. And along the way, I've made a really good research partnership with David Maya, who finding research partners and collaborators and treating them as partners is really important. And so we're the same age, sort of same career trajectory along the way. And when we first started with David May and I and our team first started, or before we started, the typical survival of cryptococcal meningitis in HIV population was over 50% mortality by 2 weeks. And so that gave us a lot of room to work with, I guess we'll say. And that if the outcomes were perfect, then it would be very hard to improve upon that. But fortunately, we have been able to improve upon that. Really over the last 15 years, such that in our current study, our 2-week survival is 95%. And so that's like amazingly good. And so I'm not sure that would have been possible. I would have even thought that was possible 15 years ago. But we've sort of been working on this over the last several decades. And so what do I actually do? And so basically, there's 3 fundamental questions that we have tried to answer. And so basically, how do you diagnose meningitis? Why do people die of meningitis? And how do you decrease those deaths? And so sort of very big picture fundamental questions. And so when I think about our career and my research team, these are kind of the questions we ask. And so you kind of put that in different grants terms, but we kind of have a sort of a focus on diagnostics, and we've done a number of FDA registrational trials. And so we provided the clinical data that led to the ME Craig LFA being FDA approved, the BioFire film array sort of PCR sort of diagnostic test. We've done a number of other lateral flow assays for crypto, many of which are not very good and are not FDA approved, in part because they're not very good. But we were able to do the field trials, and as well as for TB meningitis with the Gene Expert and the Expert Ultra for WHO approval. And so a lot of diagnostics, and then focus on pathogenesis, trying to really describe why do people die. And so that the more I learn about Cryptococcus, the less I know. And so I think that's very true of other many disease processes. That the more you get into it, you realize how many questions that there are. And so we collaborate with basic scientists on sort of the microbial pathogenesis and the genetics of the microbe. We look a lot at the host immunology, looking at just CSF cytokines, flow cytometry, characterizing the cellular population in CSF and other proteomic sort of analyses. And then look at outcomes. And so looking at how do you improve survival, through clinical trials, looking at just observational epidemiology and what are the risk factors for mortality and trying to address those, and how do you improve medical care in resource limited areas. And so one of our probably biggest sort of things we stumbled upon is how with amphodeoxycholine you get a massive amount of hypokalemia, and so replacing potassium and IV fluid management is really important in resource limited areas to improve survival. And so moving a lot of the sort of observations we make into WHO guidelines and national guidelines to sort of improve care. And a number of sub-projects along the way. And so we've primarily focused on Cryptococcus, looking at the asymptomatic Craig positive population to prevent disease. And now we look at TB meningitis now that we can diagnose that a little bit. And so we took the plunge a number of years ago on sort of the clinical and translational research and how to do that. And so initially, as a junior, faculty, as an ID fellow, we started out with a lot of descriptive epidemiology projects. So just like what was happening with the patients, and we focused on immune reconstitution syndrome, which at the time was sort of a new thing or newly described thing. And so looking at like how do you define it? What are the case definitions? Looking at pathogenesis and observational prospective cohorts. And then we sort of moved towards interventional trials after we had some preliminary data and some hypotheses we could look at. And so a number of interventional trials and meningitis diagnostics. And so along the way, I guess with the progression of funding, I guess I would sort of categorize sort of, early, mid, and later funding. And so initially, when you look back, I was a fellow between 2005 and 2007. There's some institutional startup funds, some philanthropic sort of donations. I was on a T32, which doesn't get you a whole, doesn't have a whole lot of research funds. But we were able to sort of do a prospective cohort study and some basic stuff, based on that startup funding. And then eventually I got a K grant. It did take me 3 times to actually get a K grant over about a 2-year time period. But I was on an institutional, what was a KL12 at the time, or now is a KL2 award. And then eventually we got an R34 clinical trial planning grant, and I got an R21 to do some neurocognitive function outcomes. And then we had a U01, which was looking at timing of antiretroviral therapy after crypto, which is the COAT trial, which sort of showed the delayed HIV therapy was superior. And then along the way, we really have accumulated a lot of great, which were at times students or residents or fellows who now are all faculty, and they've gone on to develop their own sort of K funding and their own sort of direction in their career trajectory, which has been great. And so our ongoing trials, we have a number of trials that are looking at sort of crypto, looking at sort of prevention, and looking at TB meningitis, and sort of the topics. These are sort of various topics of sort of, different formulations of drugs, new drugs, and different dosing. And I think also diversity of funding sources. And so while we do have Niad funding, we also have NINDS funding because we're looking at meningitis. We have European funding and UK funding as well, run through collaborators in Africa and the UK as well. And so a diversity of funding sources to try to answer clinical questions. And so some of my advice for just grant applications. So if you have a good idea, eventually it will be funded. So, for my K award, it took me 3 times to get a K award. Some of the projects we've had have taken multiple years to get funded. So don't give up. So, in baseball, a 300, 30%, batting average is you're an all-star. And so I think that's very true with NIH grants as well, that you're going to get rejected. You're probably going to get rejected more times than you are going to be successful. But if you keep trying and you have a good idea, then continue to apply and reapply and make your applications better and better. And so I think that is one of the lessons that I have, and that's easier said than done. But keep applying. Collaborate, I think, is another perspective. As a junior person, if you don't have grants, collaborating with more senior people that might have more experience can be helpful. Similarly, as a senior person, collaborate with junior people because they often have more time than a senior person does actually to get stuff done. I think one really critical advice that I didn't really appreciate early on was really talking to your program officers. And so while you can email them and request stuff, like sometimes program officers will tell you things over the phone that they will not put in writing. And so I think that's very true of the program officers. And so they'll be kind of, some of them who are really good will be very frank with you and give you some very good advice, but things that they may not wish to have in writing. And so ask for phone calls with them and talk to them because you may get some more blunt advice that way. And as Dr. Love mentioned, work on your specific aims page early on before you start writing your entire grant, and get feedback on that. Get feedback from your colleagues. Get feedback from people in the field, outside of the field. To ask an external colleague to review a 1-page application that's not a big ask. To review a 100-page grant is a bigger ask. But get that feedback early on because if you have a good focus of where you're going with your specific aims, then the grant writes itself somewhat. Last, I'd say everything counts in a grant app. So if you have grammar problems, then that's a problem. So when as a reviewer, if I'm seeing grammatical incorrectness and just sort of sloppiness, I kind of think about is that how people's research goes as well. And so I would say everything counts stylistically. Make your grants pretty and easy to read. And so while you can technically fit the 0.5 half-inch margins, and you can fit the maximal amount of text possible on a page, a reviewer may not really kind of respond to that very well. And so making sure it's easy to read because you don't want your reviewers to hate you after they open up your grant application and look at the first page and like, oh my gosh, I've got another 100 pages of this. And so do make it legible and easy to read for your reviewers. And then lastly, I think, especially when you talk about clinical stuff, the innovation criteria is often incorrectly reviewed. And so I say this having been on a study section for the last 4 or 5 years, and that it's hard for people who are not clinicians they don't know about clinical innovation. And so if you have something that's going to change the international, US or WHO guidelines and be very clinically innovative, you need to overtly state that. And so the NIH definition of innovation is below there, and talks about shifting current research or clinical practice paradigms. And so a lot of times reviewers might focus on the methods of are you using some new shiny tool, some clever methodology that's new and novel. But sometimes like a randomized clinical trial, there's nothing innovative about that research design, that research method, but you're answering a clinical, you may change the clinical practice paradigm. And so sometimes spelling out what that innovation is, particularly if you have a very clinical grant, can be helpful of spelling out what the definition of innovation is for the NIH reviewers, because sometimes that is reviewed incorrectly, which is kind of annoying. And so you can be annoyed by it, but trying to fix that up front can be helpful. So that's one of my advice for clinical applications. I really have a large appreciation for team science, and so there's a lot of great collaborators we have over the years, some of which are branching off in their own career paths now, but all across the world, and very appreciative of everyone, some of whom are on this call as well. So we have a great team. Here's a picture of our team. It's a very diverse, multinational, multi-continental team. And lastly, for those who are interested in Cryptococcus, or maybe you're not interested in Cryptococcus, but you should be interested in Cryptococcus, there is the ICC meeting, which is going to be in January 2022 after a slight delay due to something called COVID. But it's a really nice meeting. It's going to be sort of a 5-day meeting held in Kampala. And coming from Minnesota, Uganda is really nice in January. And that's true of most places in the US. So thank you very much for your time. And so I think we now open up sort of to discussion and questions and answers for the 4 panelists.
Yeah, I think so. David, I think it's Tom and I, since we're tag teaming. No one's tag teamed yet since Pete's done it all.
Well, I'm on here until David gets on. I'm on here until Tom gets on.
Oh, he's on. I see.
Oh, there we are. Okay.
So,
Tom, are you going to give us the finger?
Yeah, exactly.
So Tom, do you want to start out asking questions, or do we have anybody in the audience in the chat box want to talk to anybody about these actually very insightful discussions? Trust me, I've been in this business a long time, and all 4 had very important issues to say.
Yeah, I would really agree. These are some 4 superstars of clinical translational research, so it's your opportunity to ask them questions. Let me ask one to start for Mihalis, maybe. And so there may be people that are listening that aren't answering this, but think, oh my gosh, how could I really interact with him? Is he interested in my either clinical material, my clinical expertise, my clinical problems that might lead to some of these novel discoveries, or even from a basic science? Sort of describe practically, how would someone interact with you and your team at NIH in the Clinical Center?
I think, it's as easy as dropping an email, and we're on our iPhones more than what we should be, but I think that's really the easiest way, and we're very easy to approach, and we always respond. So I think whether it's a clinical idea, or whether it's a patient, or whether it's a basic idea that somebody needs some, advice or samples or input, just an email will be enough.
So you would encourage sort of basic scientists, but also clinicians to
absolutely
reach out? Yeah,
absolutely.
While we're waiting on other questions, I'm going to follow up on that too with Mihalis, since we've had some nice interactions over the last few years. Can you give some appreciation to the intramural program? For one of the things I've noticed about NIH is they are extraordinarily good on animal models and the ability to make mutants and stuff like that, mutant strains. For people out there trying to interact with you and stuff like that, is this an entry point that they could talk to you or interact with you for mice strains and things like that, where really it seems like NIH is really well set up for that? And the second question, while you're thinking about that one, is in this day and age, you talk about very interesting cases that are unique, like the patients you showed us and stuff like that. And those are great to bring to NIH if you can get them there. But what about the issue now with genome sequencing and stuff like that, that really what we out in the hitherlands need to do is to be able to capture patients, get DNA off these patients, get clinical information off of them. Is there a vision where you could see us sending you sequence information, or at least DNA, and that could then be utilized by your group and your bioinformatics and stuff like that?
So I will just kickstart it, and then I think Steve can even give a bigger overview of some of the Nyad resources that are aimed to intramural investigators, but they're certainly through collaboration available to extramural collaborators as well. So I think we do have core facilities where we could design and make mouse strains. I think these are available not exclusively in intramural Niad or in intramural NIH, but in most institutions. But there are ways by which we could generate mice that might be at the core of a collaboration between intramural and extramural scientists. So I think from that standpoint, we do have those means, and I can think of examples where my group, Steve's group with Coxie and other groups have interacted with that regard, making mice to support mechanistic studies. With regards to patients, I think, John, we're really open to all possibilities. I think, we love to see the patients here. As Steve said earlier, there's really no much of restriction outside of, the person willing and able to kind of visit the not so cold as Minnesota kind of DC. So that we always welcome seeing patients and helping in that regard. In patients where that's not feasible, or if there are investigators or clinician investigators that already have a biobank of DNA samples, like, for example, how our collaboration through the years has been fruitful, we pose a mechanistic question, and we ask the question, how does that mouse finding relate to humans, if it does? And in that regard, we didn't have to, reinvent the wheel and get to do what took you so long to do astutely, but rather collaborate with you and get information, whether that was done in your lab or could be done in our labs here through MTAs that can, allow transfer of material. I think there are multiple possibilities, and we're open to all of them. And some cases might be different than others with regards to, how the samples might have been collected in XYZ institution or whatnot. But I don't think there is really much of a restriction with regards to how we can all work together to get more information. I would finish by saying, and I think you said that over a decade ago, when that was not even a sexy topic. I think every patient, in a way, even not only those very extreme examples that we showcased, but every patient who gets into the ICU, for example, and gets a mold or a yeast infection, they have likely genetic or immunological modifiers that make them develop more or less severe disease. And we just in ID have not yet reached the point, and in clinical mycology haven't yet reached the point to be able to pinpoint those the way that our hematology and oncology colleagues are able to really do and even implement targeted therapies. But I think that's where we need to go. And that cannot happen only with one way. I think taking advantage of all of the resources and all of the collaborative efforts, I think will get us there.
Can I make a quick point, Mihaly? I'm sorry I missed most of it because I was giving another talk, but I think one of the very difficult things for us at MD Anderson is longitudinal sequential effect of immunosuppression. For example, if you ask the question for how long somebody will get ibrutinib and then have had steroids before or gets another monoclonal antibody afterwards, and disease relapses, blah, 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 how you longitudinally follow up this patient, which is unique and an outlier. So sending a patient as, some serum or some material to NIH is important, but I think maybe we're missing the opportunities of longitudinal assessment of this, and it's not an easy way to do it because we cannot send this patient to NIH for ever, and we don't have the infrastructure necessarily to follow up a patient with a weird fungus in the brain who got A, B, C, and D, or B, C, and D, or C, D, and how you assess these outliers with this dynamic interrelated immunosuppression to study them comprehensively.
I think, for some of those settings, I would answer, more broadly to say that, some of those studies likely will need to be done locally. If you're talking about, immunological assessment in neutrophils, for example, that cannot be done outside of, let's say, MD Anderson. But we're open to, share our expertise on how somebody can take neutrophils and, look at the million readouts where you are through, even joint grant efforts. And, the other thing that, we're very cheap dates here being on grants because we don't get any money. So I think I would say it's impossible to assess that by bringing these patients here, many of whom are sick enough that may not be able to travel, but there are ways to share expertise and set up collaborative projects that can be done in every other place as well as we can do it here to some extent. So
one of the questions in the chat is it possible for students, postdocs to come to a lab at NIH and work? Maybe not in COVID, right? But Steve, you want to take that?
Yeah. No, we're we got a whole fence around this place here. Once we get them in, they never get out. We love it. It's the only way we get fresh flesh here. We need new people. So students can come in, absolutely, and each of our laboratories, if you wanted to come to Mahalis's lab, you'd work through him directly. If you're talking about students that want to do rotations on medical electives, we've got that. Many different opportunities for students, even for training periods. That's easy. If you want to come for 1 year or 2 years, those are also available. We're very, very student friendly, despite the fact that we don't have a direct affiliation with a medical school or a university like many of our peers do. Always interested.
Yeah, Steve, I would say that it's true that they stay there a long time once they get there. I think Dr. Fauci may be the poster child of that.
He might be.
I imagine all of you are on Donna's listserv too, but she would add you if you want to be. She will put in her weekly emails to everyone, multiple training opportunities, courses, lecture series and stuff. So that's also a good source.
That'd be great.
You want to add anything, Donna, to that? Are you still on?
Yeah, sure. I was just going to type in the chat that please shoot me an email and I'm happy to add you to the listserv that I send out periodically with opportunities. And likewise, if folks have opportunities in their lab, I'm happy to send those out as well. But I've been noticing also that Twitter is quite active and people post tons of job opportunities there. So for folks that aren't on sort of science Twitter, I would encourage you all to do that.
I would just, I see Erica's posting a clarification about, somebody could come in a lab and for a few weeks to learn a methodology. So I can just, I don't know if, I don't think Mark Sweetergirl is still on the line. Otherwise, if he is, he should, say it himself. I think he just stepped out. But, Mark emailed me maybe a year and a half ago that he wanted to learn how to make, do bone marrow transplants in mice for his own project. And he came in our lab for a month. We had coordinated and set up the timing so that he sits and learns. And now he does transplants in California with a better weather and, without us needing to be there to. So absolutely yes is the answer.
There were a couple questions about figuring out which study section the application could go to. Dave, you want to?
I mean, I think overall, like worrying about which study section things are going to go to, I wouldn't worry about it. It's sort of, it's going to get assigned someplace, and they, they reorganize these every couple of years, and so I think it's sort of an exercise in futility for the most part. That it's probably going to go where it's going to go. And even if you request it to go somewhere else, CSR may not even follow that request.
Okay, Donna has put her email in the chat if you want to be added to her list, so that'd be good.
Tom, can I ask a question?
Absolutely.
Sure. This is Pete Pappas. One thing that seems that we have underutilized. It's easy enough, perhaps, for individuals to reach out to our intramural colleagues, send samples, request service, ask advice, send patients. They're terrific about that. But as a group, since many of the people on this call are MSG participants, one of the things that we haven't done is to collaborate, other than to use the administrative services of the NIAD. In other words, we for all the large studies we've done, we've never volitionally collected samples for immunology or genetic purposes and so forth, and then provided them to our colleagues, our intramural colleagues, to help enrich a study. And I don't know the reason for that specifically, but is there any reason that, I guess my question is this, is there any reason that an intramural individual can not be included in a extramural type of application? I mean, it sounds like a conflict, and so I'm just wondering how that works. If, in other words, if the group were to submit, let's say, a candidemia study or Canada in the ICU, and we had beaucoup samples on patients in the ICU, but we wanted to use Mihalis as a resource within. How does that occur? How does that occur at a grant level? I mean, can Mihalis help on that? Is he excluded because he's an intramural guy? Or does he help us write it? I mean, clearly there's some things that his group can do that not many other people anywhere can do. So I'm just throwing it out as
I think you've already done that, Mihalis, haven't you? I mean,
Yeah.
the U01 grants actually do exist to do exactly this sort of thing. We have one right now with John Galgiani, George Thompson,
Okay.
where
Okay.
samples are collected and sent in. And the only thing that can't go back and forth between intramural and extramural is money. Everything else can go back and forth just fine.
But as far as, but I mean, you're acting more than a service center. I mean, you're providing intellectual input into the grant application.
Well, you're the first one who's ever implied that, but I'm grateful. And, yeah, no, it's entirely doable.
Yeah, and
Donna could correct me if I'm wrong, but we can be collaborators on R01 grants. We just can't take any money.
Right.
That's absolutely correct. Yeah.
I have been a collaborator in certain R1 grants, and the only thing we cannot get is financial support, but we can provide the expertise, collaborate, do experiments gratis. So that's the only,
I'm surprised that this doesn't happen more often. I really am.
So are we.
Yeah, I'm very surprised it doesn't.
And we're very receptive.
Okay.
And the U01 mechanism that some people have been involved with, the U01s, which can be either entirely extramural or collaborative between the clinical center and the extramural community, we can get funding through another mechanism, it doesn't come out of your part of the grant, but there are ways to make sure that if there were a substantial effort that had to be undertaken to do something with samples, for instance, that could be funded. So there are mechanisms that can be used, and they should be pursued. We're very open to it.
Thank you.
I have a question for David. We've got a couple more minutes, and he hasn't said anything here. David, with your translational material and some of the outstanding work that you have going forward to try to get it into the clinics, Donna and Bayou came up with a lot of issues that NIH actually have. They have access to all these different things, but what's your impression? I mean, to get access to chemistries, to get access to the speed of development of these things, is an SBIR route or having a partner in industry helpful or not?
So yeah, I mean, for the SBIR, I mean, it requires an industry partner, although, so we created our own sort of spinoff for that. I mean, that being said, just I think in the last month, Donna and Baoying and I talked about, for example, studies to get Turbine Mycin moving forward, and they identified a number of opportunities that in fact don't require industry partners. And you mentioned the preclinical resources, and those are really excellent. And as I said, they're in kind. So I think there's a lot of opportunities to fund these translational things that don't require industry partners, which is fortunate because, as a lot of you know, it's getting harder and harder to get those industry partners given the financial constraints.
Yeah, thanks, David. I think you're right. One quick question for David Boulware before we leave. David, for the investigators getting up and trying to deal with clinical research in the United States, is it another leg to deal with clinical research internationally and with NIH and all the lawyers and everything that go on? What's the strategy, or is there concerns about that?
Well, if you can avoid lawyers, that's always the best. That's a general advice. You know, I think it depends on your institution and how savvy your institution is working with international collaborators. Ultimately, for collaborators, it's really about a partnership. But you know, there's ways to move money. There's you know, standard subcontracting procedures, and so it's a little bit more challenging internationally based on the partner, and so how strong that partner is financially. Do you need to front the money? You know, can they invoice you? You know, and so there are some additional issues, but Fogarty provides some advice on how to deal with international contractual arrangements versus in the U.S. And so I don't do a whole lot of U.S. research because there's just a different amount of contractual paperwork that gets involved on the U.S. side and institutions. So I don't think internationally it's not, you know, it's just different. But yeah, there's there's multiple layers of regulatory approval, and so things are a little bit slower, but it's possible.
But I think for everybody doing this thing and thinking about international or national, they should get their grants people involved and their lawyers involved early rather than later.
Correct. And I think, you know, it's something not working in a vacuum. And so if you have a mentor or other people that work in country or asking other people for advice, you know, so you're not having to reinvent the wheel, because, you know, some of the same issues that come up for a new investigator, you know, maybe I've dealt with 15 years ago. And so, you know, asking around can be helpful as well. And I would say also for those interested in international research, you know, particularly for fungal diseases, you know, the Fogarty program, you know, the Fogarty Fellowship Program that takes postdocs, there's a number of consortium institutions, and they take people from anywhere. So you don't have to be part of the consortium, you just have to work with the consortium. And so, you know, we've taken postdocs not from, you know, we have a consortium with Minnesota and Michigan and University of Washington and Hawaii, but we've taken people not from our consortium to work on projects. And so I think that's one postdoc opportunity if you're a graduating, you know, ID fellow and you don't know exactly what you want to do. You know, there's a number of people on this call that have done Fogarty fellowships over the years and have changed their career paths.
Thank you. Tom, do you have any other questions or anything else?
No, I think it's been a very exciting session. I really encourage the listeners the viewers to really take advantage of these. I'm agreeing with Pete. I don't know that we really involve these guys in a lot of the things, and girls and things that, you know, the extramural work that we do. And it's a tremendous, you know, opportunity. Like Donna showed, we're involved in the preclinical services work, and it may be kind of the extramural stuff is like when it's from the government and it's free, it sounds too good to be true and people can't deal with it. But it is, so there you go.
So I appreciate all the messages from all the people that have talked, and I think for young people or other people on this conference, please take advantage of these things. Talk to people, talk at NIH, talk to collaborators, connections. There's a lot of resources there that really are not utilized as well as they could be. And if they can be, you can really go out and do some very, very interesting things. There's a lot of tools out there. Which I think brings me to try to keep on time. And I do thank the speakers. I have been enlisted to be the convener for the next session, which is an interesting session, which I don't think ever has been done before. And I don't know how it will actually work. I've had some pretty seasoned investigators up here that know how to talk and end up in 10 minutes and stuff like that. And nothing against these young investigators, but it's been probably early for them in their presentations. And we're going to do some lightning talks on future innovations for translational science in medical mycology. And we searched out and found 7 young investigators, and they're listed in the program. And I'm actually going to ask them to go from one to the next and introduce themselves and where they're from, and spend their 5 minutes doing what they want to do to show a little bit about their translational science. At the end of the time, please keep up your questions and things, and we'll go back and try to interact with them.
Thank you for inviting me to speak. I'm sharing my talk right now. Please let me know if you can't see it. So my name's Jeff Jenks. I grew up in Ohio. I'm currently living in San Diego, where it's raining cats and dogs here, which is unusual for this time of year. And I wanted to talk more about a research topic that I'm interested in, but haven't embarked on, with hopes of maybe getting some feedback from all the experts on this call here. So there's people on this call that know the immunology a lot better than I do, but just briefly, regarding the immune response against Aspergillus, when Canidia or hyphae make their way into the respiratory tract down to the alveoli, some of the initial cells to respond to these pathogens are alveolar macrophages and neutrophils, and then after the innate response, the adaptive response kicks in, and these antigen-presenting dendritic cells present these antigens to naive CD4 T cells, which release cytokines, kind of recruiting these T helper cells, and then other cytokines are released. And this includes IL-6, IL-8, IL-17, 23, and others. And so what the research with, or some research with these infections has shown is that IL-6 and IL-8 are immunologic markers elevated during infection, invasive aspergillosis infection, as well as with other molds, and IL-17 and 23 markers that can be elevated with IA, as well as invasive candidiasis. So I just pulled a table and some figures from a couple papers. So you can see here in this table of BALF that IL-8 was significantly associated with differentiating between probable IA versus no IA. And from serum, both IL-6 and IL-8 could differentiate between probable IA, no IA. And then here's another paper with these two figures that look at trending these inflammatory markers. And so, even within 4 days after bronchoscopy, uptrending IL-6 and IL-8 markers from plasma was associated with poor outcomes and death in these patients. So one topic that I've thought about is, so these studies have looked at using these immunologic markers to diagnose infection after symptoms have developed, or there's signs on imaging of a invasive fungal infection or candidiasis. But could these immunologic markers be leveraged either with traditional biomarkers or alone, and diagnose an infection early before patients have a chance to develop symptoms or there's findings on imaging. And so here you can see kind of the frequency of our lab testing here at UCSD in solid organ transplant patients or patients who have undergone allogeneic bone marrow transplant. You know, the labs are gotten pretty frequently. And so, you know, one idea I've thought of is getting extra serum or plasma on these patients, you know, when they get their routine lab draws, storing this, and then, you know, down the line, a year and a half later, looking at these immunologic markers in patients who have gone on to develop invasive fungal infection and comparing those levels to a subset of patients who either didn't develop invasive fungal infection or develop infections from other pathogens, viruses, and bacteria, and to see if these immunologic markers are associated with invasive fungal infection, and if increased levels are seen earlier before patients have a chance to develop symptoms or have signs of infection on imaging. So this is the basic kind of premise of you know, this study. You know, so that's basically kind of one of my research ideas, and I'm happy to turn this over to Julie. I'll get out of here.
Hello. Hopefully everybody, thanks for the introduction there. Hopefully everyone can see my slides. I'm actually going to stick with the theme of diagnosing fungal infections with a slightly different approach. I thank you for giving me the opportunity to speak today. I'm Julie Steinbrink. I'm a T32 Transplant Infectious Diseases Fellow at Duke University, where I'll be coming on as faculty in July. And so my research is looking at host transcriptomic biomarkers as a diagnostic tool, particularly as it relates to fungal infection. So first of all, what do I mean when I'm talking about host transcriptomic biomarkers? Well, traditionally in infectious diseases, if we're trying to determine what someone is infected with, we do this often by direct pathogen identification methods. However, through a host biomarker approach, we can instead look at the host's response to a particular pathogen. And since each particular pathogen elicits a specific immune response that's unique to that pathogen class, we can examine and analyze that immune response by how it's reflected in its respective genomic response. And we can do this by looking at gene expression data in the peripheral blood. Now, our group at Duke has done previous work with using these gene expression-based classifiers and biomarkers to diagnose viral and bacterial infection with a high degree of accuracy. In order to further grow this work, I wanted to use it for the purposes of fungal infection. And additionally, since a lot of the patients that are often at risk for fungal infection are those that have inherent forms of immunocompromise, such as transplant recipients, since this particular diagnostic approach requires the host response to a particular type of infection, can this approach still be used for these patients who by their very nature have an impaired host response? And so I've had success working with two major fungal systems. The first is a mouse model of Aspergillus, and the second is a human model of Candidemia. In the first model, we were able to generate a 146 gene classifier that was able to discriminate between infected and uninfected mice with invasive aspergillosis with a high level of accuracy. However, when the mice were exposed to immunosuppressive medications, they exhibited a confounding effect on the performance of the classifier, and the classifier could no longer as accurately diagnose between the infected and uninfected mice. Once we took this into consideration and we controlled for the genomic effects of the immunosuppression, we were then able to generate a new 187 gene classifier that could perform well in both immunocompromised and immunocompetent states. And that's demonstrated here by this violin plot, where you've got your predicted and observed infection statuses, and you can see that the model accurately predicts the infected and unaffected status between the various levels of immunosuppression. Therefore, demonstrating that when appropriately designed, with the immunosuppression taken into account, these host-derived transcriptomic responses can still accurately diagnose Aspergillus infection, even when the immunosuppression is present. In the second model, we looked at hospitalized patients with candidemia, as well as comparator groups with acute viral, bacterial, non-infectious illnesses, and healthy controls. And from this, we were able to generate a 94 gene signature that could accurately distinguish between all infection phenotypes with a high degree of accuracy. And you can see that here with the AUC curves on the top, as well as the respective AUC values for each phenotype. And on the bottom, we have a box plot with the predictive probability of infection for each particular infection phenotype as well. So this also demonstrates that this is a promising diagnostic approach for simultaneously differentiating between multiple types of clinical illness in at-risk acutely ill patients. And this is really great because this is representative of what we would see in clinical practice, where you have someone presenting with an undifferentiated febrile illness in your emergency room, and you're trying to figure out what the etiology of that illness may be, and so therefore has direct clinical applications. Now, a lot of this initial work has been more in the discovery phase, but however, for future research directions, I'm trying to plan to pursue the translational aspects of this work and its application to clinical practice. So for the first infection model with Aspergillus, since I plan to use the same approaches that were used in the mouse model to generate a transcriptomic signature of aspergillosis in the human model as well, and assessing its performance in the presence of immunosuppression in this group. Now, for the candidemia model, we already have human data, and I want to continue to work on optimizing and simplifying this gene signature into a size and format that's more translatable to a clinical point-of-care platform. Our group has some experience using such platforms. We've published our experience putting viral signatures on the Biofilm array system. However, this has only about 45 targets. And the signatures that I've described to you here are quite large. We've got about 187 genes for Aspergillus and 94 genes for candidemia. So currently, both of those are too large and complex to directly apply to clinical care. But through some of our early further analysis for the candidemia work, we've found that that signature for the candidemia has the potential to be reduced down to about 23 to 26 genes, which would fit into that target without significant loss of test performance. So this highlights how near translational this work could be and its exciting future potential in the non-invasive diagnostic space. And so with that, I will stop and let the next speaker go on, but I'll be happy to take on questions at the end. Thank you.
All right. Good afternoon, everybody. I'm Todd McCarty. I'm an assistant professor in infectious diseases at UAB with Pete Pappas. And in the words of Monty Python, now for something completely different. Mine will be more on the kind of clinical epidemiological end of sort of translational ideas. And these are not all uniquely my own. And sort of having Pete Pappas down the hall have that opportunity to have had discussions around this sort of thing and participate in the original Transnet. And so that epidemiologic study was across 23 transplant centers throughout the US. And it was a nice and unique prospective observational surveillance study encompassing 6 years with both solid organ and stem cell transplant patients. And to date has produced 10 manuscripts, and you see the array of different categories that the data has kind of touched on and been very informative, and I think driving a lot of additional research moving forward. And so I think there are a number of different reasons from an epidemiologic standpoint to repeat a similar Transnet type of study. You see sort of rising invasive infection rates and susceptible groups that are evolving over the sort of spanning time since that study finished. There have been new treatment agents, new prophylactic regimens, new practices, anywhere from monotherapy to more combination therapy, not to mention sort of the difficult task of therapeutic drug monitoring and the variety that you get from center to center. There have been noted changing resistance patterns. I think the biggest one being Candida glabrata with a kind of echinocandin resistance, but certainly the introduction of Candida auris can't be understated, and the sort of looming specter of azole-resistant aspergillus is one to always kind of keep an eye on. And the taxonomy is certainly keeping everybody busy with the identification of new species and subspecies and species complex, and how best can we kind of understand how those differences in different organisms play into clinical disease, and using a large sort of platform like this to better understand, are there important differences or are they clinically identical syndromes? And lastly, we have sort of newer and enlarging patient populations that are already happening, but also potential new patient populations moving forward. And so we've seen a big increase in HIV transplantation, and more recently Hep C, donor positive recipient negative solid organ transplantation, expansion of vascular composite allografts, and now with uterine transplantation, we're seeing new organs. There is some potential, could xenotransplantation become something that is further studied in the not too distant future. And then on the sort of stem cell end of the spectrum, sort of expanding usage, expanding into a greater patient population, as well as CAR T cell therapy and immunotherapy, and best kind of understanding where is the greatest risk, how do these patients present, what sort of are their unique avenues to how do they need to be managed and diagnosed. And on this sort of end, in the midst of doing this, ideally you can sort of evolve the study and use more, while there was a lot of sort of specimen banking and thinking about genetic study into the pathogens, there wasn't as much of an opportunity for investigating the patients themselves. And I think some of the earlier speakers have talked a lot about understanding why it is an individual does get an invasive fungal infection beyond just they have a transplant and they're on some immunosuppressive medicines, because it is still an uncommon and sometimes rare diagnosis. Could you add a layer of patient plasma and serum banking to better understand some of the prior mentioned immunologic profiles to better understand how to assess risk and diagnosis? Are we seeing more geographical shifts in some of these agents that have in the past been more sort of restricted or understood to be restricted? I think we're seeing with influenza-associated aspergillus and SARS-CoV-2 associated aspergillus, the Kappa, can we sort of better understand some linkage to other precursor infections as well as those? Could this be a platform for tracking how novel diagnostic and treatment regimens are being implemented, as well as some of the more sort of novel diagnostic mechanisms? One of our biggest challenges in this patient population is always, there have a lot of potential things, not just fungi, that could be causing their illness, but they're not well enough or too complex to get tissue, and we are left kind of stuck trying to make a diagnosis in other means. And so with this sort of specimen plasma banking, is there an opportunity to loop in next generation sequencing of both plasma and tissue, other whole blood PCR mechanisms? I think it would be sort of a nice, again, broad national platform to link together a lot of different centers and have a lot of data and specimens banked for the future and continued sort of understanding of an evolving patient population. And I will hand it off to Shannon Escher is up next.
Okay. I hope that everyone can see my screen and hear me. My name is Shannon Escher, and I am a postdoc at Tulane University in Dr. Mari Nover's lab, and I'm very excited to have this opportunity to share my work with you all. So in the Nover lab, we are broadly interested in mechanisms of immunomodulation and systemic candidiasis. And in particular, we've spent a lot of our attention on intra-abdominal infections, which have a very high morbidity and mortality. And these infections are often polymicrobial. And in particular, when fungi are involved, their mortality is exacerbated. So several years ago, we developed a mouse model of polymicrobial intra-abdominal infection with Candida albicans and Staphylococcus aureus, in which when we inoculate the mice intraperitoneally with Candida or Staph alone, they generally survive the infection. But if we inoculate them with Candida and Staph together, the mice die very quickly. And we've shown that this mortality is due to a robust inflammation and pro-inflammatory cytokine production, indicative of lethal sepsis. And so we began investigating other non-albicans Candida species, and we discovered that low virulence Candida species, in particular Candida dubliniensis or C. dub, actually did not share this synergistic lethality. And in fact, if we inoculated mice with C. dub prior to lethal co-infection, we could protect them against sepsis. And so we became interested in how C. dub was actually inducing this protection. And through a number of studies that I won't go into, we have found that protection is actually not dependent on adaptive immunity or on macrophages, but rather that it's dependent on GR1 positive cells. And so the most common cell types that express GR1 are neutrophils and myeloid-derived suppressor cells. And we actually believe that the GR1 positive cells that are mediating protection in our model are these MDSCs. So what are MDSCs? They're a heterogeneous population of immature myeloid cells that share common biological activity of suppressing immune responses. They co-express GR1 and CD11b. And while they're most well known for their role in cancer, they've also been shown to have a role in reducing sepsis. And so these cells are developed through a two-step process. The first step is the expansion of the myeloid compartment in the bone marrow. And then the second step is the pathological activation of these immature cells into mature MDSCs that then express their suppressor functions. And so we're hypothesizing that C. dub is inducing this expansion step in the bone marrow, and then lethal co-infection induces the pathological activation of these cells. And so I'll just tell you a little bit about my work on looking at C. dub induction in the bone marrow. So we've previously reported that C. dub can actually access the bone marrow compartment pretty quickly after intraperitoneal inoculation. And we've actually shown that C. dub infiltrates the bone marrow at a level that is higher than Candida albicans or Saccharomyces or other non-albican species that we've tested. And in fact, we've shown that the level of bone marrow infiltration actually correlates with the level of protection provided in our intra-abdominal model by these different Candida species. And so when I joined the lab, I became interested in actually what is happening in the bone marrow. And I started by looking at the specific cell populations that reside in the bone marrow. And what I found was that the total hematopoietic stem and progenitor cell compartment was actually expanded within 24 hours after C. dub intraperitoneal inoculation. I also found that the myeloid biased progenitor cells were expanded at the expense of lymphoid biased progenitor cells. I found a rapid increase in granulocyte macrophage progenitors and also an increase in putative MDSCs. And so all of this data together really suggested that C. dub is inducing a bias towards myelopoiesis in the bone marrow. I also found that if we block hematopoietic stem cell expansion using an antibody that depletes these cells, we can actually abrogate protection in our intra-abdominal model, really indicating that this stem cell expansion step is important for protection in our model. And so in summary, we believe that this is a novel form of trained innate immunity, which we have termed trained tolerogenic immunity, that limits inflammation and sepsis via trained MDSCs that originate in the bone marrow. I've shown you that C. dub can induce the expansion of these myeloid cells in the bone marrow, and we believe that secondary stimulus with lethal co-infection can activate these cells. My ongoing work is focused on actually identifying the mechanism of how C. dub is inducing or how C. dub is training stem cells in the bone marrow, as well as actually characterizing the function of mature MDSCs during protection against lethal sepsis. And with that, I'll acknowledge my lab and our funding sources and thank everyone for coming and giving me this opportunity to share my work. I look forward to our further discussion.
Good afternoon, everyone, and thank you for this opportunity and allowing me to share our recent results with you. I'm Fariba Donovan. I'm part of the faculty here at the University of Arizona Infectious Disease and Valley Fever Center for Excellence. And I'd like to talk to you about the biologic response modifiers and their role in increasing the risk of coccidioidomycosis. As you know, there are increasing number of biologics that are being used recently in autoimmune disease such as rheumatoid arthritis, and so it causes further risk for endemic mycosis, and specifically in our area, posing increased risk for valley fever. Some patients who are on biologics, we can see here that they can handle coccidioidomycosis very poorly. And it has been already reported that the risk of disseminated cocci goes up to almost 150 folds in some of the patients who are immunocompromised, such as those who are on biologics. So the very immediate questions that we face every day here in the clinic is it really safe to use these biologics in an area like ours that is endemic for valley fever? So to answer these questions and consider translational nature of this study, we designed this animal model using the laser pointer here. We use this animal model that is a B62F1 mice, which is intrinsically resistant to valley fever. And we use a strain of cocci, which is 1038, although virulent, but the progression of disease in animals is very slow. Thereby, we can actually study the pathology. So in this model, we infect the mice intranasally with 50 spores of this strain on day -1. And on day 1, we give these animals anti-TNF antibody. One group receives it up to day 14, and the other group all the way through day 70. And this is the isotype control group. And as you see here, we can see that early TNF blockade has deleterious effect on the mice survival. Now, in addition to our animal study, we are very appreciative of our collaboration with Dr. Holland and Amy Hsu in NIH, that we actually have been able to show that the disseminated cocci is frequently the consequence of dysregulated or delayed innate immune response to initial infection. Considering these both aspects, we started evaluating the CM risk in patients who are on BRM. And to do so, we started initially with a 2-year retrospective cohort to study patients who are over age 18 by reviewing the electronic medical record using the ICD codes for autoimmune disease. We have identified about 10,000 charts, and ICD code for coccidioidomycosis, we identified 1,300 charts. This study is completed. However, the data analysis is still ongoing. And if you see here, the preliminary result shows that in those patients who have autoimmune disease and confirmed cocci, the red group, they have shorter duration of stay here in Arizona than those that are actually negative for cocci, indicating that these people actually suffer from new infection, most likely than reactivation. Now, with the same study, we have so far identified about 20 patients that have autoimmune as well as cocci, and these people are on BRM. So out of these 20 patients, about 5 of them had confirmed disseminated cocci. So this actually gives us a good number for our future study that I will get to in a second in our prospective study. Also, in this group, when we compare the disseminated versus non-disseminated, we can see that there's no significant difference in the length of a stay in Arizona. So putting all these data together, our hypothesis is to consider adaptive cellular immunity in patients who have prior infection will protect them from BRM-induced reactivation or new infection. And to test this hypothesis, one of the strategies we have is prospective study enrolling the RA patients who are about to start biologics. And this proposal actually has gone to NIH. And the good news is that our retrospective study actually supports the number that we placed in our proposal to be reviewed. In this proposal, we also like to evaluate the cell surface activation markers such as CD69 as an evidence of previous infection, also considering evaluation of some other cytokines. And to do so, we like to identify the coccidioidomycosis immunity and potential alterations that may happen when patients go on biologic response modifiers. And to that end, I really appreciate this opportunity, and I'll be available for any question after the discussion. Thank you.
All right, good afternoon. I'm Stephanie Smith from Northwestern University. I'm an assistant professor of otolaryngology and health services and outcomes research, and I am delighted to be invited to participate in this meeting, and I'm going to be giving my lightning talk on acute invasive fungal sinusitis. So acute invasive fungal sinusitis, which I will call AIFS, is this rare and aggressive but often fatal disease where we get fungal invasion that starts in the nose and sinuses, perhaps the first interface with the environment when we breathe, and this fungus takes over, causes thrombosis, angioinvasion, and this real kind of rapid progressive tissue necrosis. Frequently, we see this over even hours to days, spreading to the eye and to brain tissue. Here we have a picture. This is a turbinate in the nose on a nasal endoscopy view with kind of this black dead sinus tissue. And here we see it extending to the hard palate. The most common organisms involved are Rhizopus, Mucor, and Aspergillus, but there is certainly global variation and exceptions. In terms of pathogenesis, we know that fungi are all ubiquitous, but they're usually unable to penetrate our epithelium, and they're controlled by our innate and adaptive immune cascades. However, immune dysfunction can alter our protective mucosal barriers. And while we see that AFS appears to involve like a scant but variable inflammatory cell reaction, the immune response and cytokine profile of AFS is really not well established. In parallel, for chronic sinusitis, which is a separate beast from acute invasive fungal sinusitis, I just want to draw your attention to kind of some of the advances that we have seen in understanding the inflammatory profile of CRS, chronic sinusitis. The Northwestern University Sinus Center, which I'm a part of, has recently contributed a lot to clarifying these endotypes, type 1, type 2, and type 3 in CRS. And here we see in type 3, this is predominantly the response we see for bacterial and fungal stimuli, predominantly kind of mediated by the Th17 response and neutrophils. We were able to clarify these endotypes at Northwestern largely through the CRS repository that Dr. Schleimer started. We have 4,000 specimens from chronic sinusitis surgical patients, as well as 1,000 non-CRS controls. And as I will mention later in this talk, I do want to start contributing acute invasive fungal sinusitis specimens to study those. In terms of epidemiology, we've heard from a couple of the other presentations that invasive fungal diseases are increasing worldwide. We're seeing more immune modifying drugs, biologics, increasing diabetes, and aging population. And recently, I've seen several alarming case reports of post-COVID-19 invasive fungal sinusitis. However, the epidemiologic data pertaining to AIFS is very scarce. Fortunately, we know this is a very rare disease, but we have not yet described kind of geographic patterns or patient demographics on a national level. In terms of treatment, it's really a three-armed approach with systemic antifungal therapy, reversal of any underlying immunocompromised state when able, and then the third pillar is surgical debridement. This involves really aggressive removal of any tissue that appears to be non-viable. We do sometimes proceed to removing the nose, removing part of the cheek, the hard palate, the eye and brain tissue on a case-by-case basis. But unfortunately, as a surgeon, it's hard to make the call and to guide our patients to make these very kind of scary big decisions on a minutes notice before going to surgery. And the papers we have available are all based on small case series, and we are not sure whether these major kind of disfiguring surgeries improve survival. Recently, out of California, a couple of studies have shown that conservative orbital debridement or retrobulbar injection of amphotericin may offer similar survival outcomes when the orbit is already involved in the invasive fungal sinusitis. This is an operative picture of a case I just did 3 weeks ago. This is a sphenoid sinus in a 60-year-old woman with a remote history of kidney and heart transplant on immunosuppression on tacrolimus. And she presented with a headache. Her nasal endoscopy at the bedside looked totally normal, but her CT was a little puzzling. So I took her to the operating room and certainly found just a small focus of invasive fungal disease right here in the lateral recess of her sphenoid sinus. I just want to give you kind of a idea of what we deal with as otolaryngologists, but this is the internal carotid, this is the internal carotid artery prominence right here. So we're, millimeters away, eggshell fragments of bone to the major blood vessel supply to the brain. The optic nerve runs here and our orbit is right here. So it's clear that disease can progress to the orbit. This patient had progression to her pterygopalatine fossa. And, we're trying to make these big management decisions based on limited evidence. In terms of outcomes, we've not really seen any progress despite modern medical and surgical management, even in 20 years. There's a lot of discrepancies pertaining to prognostic factors and overall survival, and a lot of kind of biased opinions. Justin Turner, one of my colleagues in about 2013, published the largest systematic review. This included 52 studies and a total of 800 patients, but there are really only an average of 16 patients per study to give you an idea of how small these cohort studies or case series are. Unfortunately, only about 50% of patients survive this disease, and we do know that the most significant positive predictors of survival are surgery and antifungal therapy. In terms of the future, the International Consensus Statement on Allergy and Rhinology wrote, there is a paucity of high-level evidence in the literature regarding treatment and survival of AIFS. And my research agenda is to hopefully contribute to that. One of the projects I'm planning and working on is to really advance the understanding of the epidemiology, prognostic factors, optimal diagnostic and treatment approaches and outcomes of AIFS. And I want to do that on a national level, looking at 20 years of data. Here's some preliminary data that I present here. It is unweighted, so I do have to correct this, but we do see that there are some variations with the lowest income quartile being more affected by AIFS, and we do see more cases in the South. And this has not been really written about previously. I will also plan to leverage large-scale EHR data from the Chicago area with over 20 million records from all the major academic centers in the area, as well as community centers, to really see whether the presentation, if you come in with an affected eye, does that lead to a poor prognosis? Does the time to diagnosis affect things? And does the fungal species affect it? And my second major goal and plan for research is to establish an acute invasive fungal sinusitis patient registry and biorepository. I'm very curious from my audience to see if you are already collecting specimens or using them or would like to use them. But I want to prospectively collect patient data, surgical tissue, sinonasal cells, mucus samples, and blood, bank these and build upon the existing biorepository that we have. We have all of the kind of processes in place. I'm excited to build this biorepository, and it's going to be multi-center right now. I've talked with colleagues in Texas and Colorado, and we plan to do more immunologic assays, kind of enable future studies into the pathophysiology and perhaps also better diagnostic approaches. So ultimately, my long-term goal is to fill in gaps in knowledge and inform future clinical trials to develop potentially vision and life-sparing recommendations for our patients with AIFS. And I'd like to acknowledge my wonderful team at the Northwestern Sinus and Allergy Center. And thank you for your time.
Well, thank you, everybody. Very, very impressive. First of all, impressive in your presentation, also in your science, and finally in your enthusiasm. So this paper is open for, we've got about 15 minutes or so to discuss with each one of these. Anybody you want to talk to, they're here ready to answer your questions. If you stay too silent on me, I mean, each one of them I can pin them down. So, but I don't want to. I don't want to pin them down. So I don't want to have to ask questions. So, is there anybody that would like to start this? These are really nice presentations.
Dr. Worster.
I already submitted a question in the chat for Jeff Jenks. It was my understanding from the presentation that these are unstimulated cytokine assays. However, I would imagine that these patients are pretty complex. They're co-infections, they have immunosuppressive agents. So I was wondering how these unstimulated cytokine assays would compare to an antigen-specific reaction. We are increasingly getting assays, both commercially and investigator-driven, that could facilitate such studies. So I'm interested to hear if such studies have been performed.
Not that I'm aware of. Yeah, and I think, you know, one of the challenges obviously would be trying to determine, are these immunologic markers elevated because of invasive fungal infection or other reasons, and that would be obviously important to determine, or else their utility wouldn't be that great. So this would be, I think this is kind of a topic that hasn't been really investigated much, particularly with early infection before patients have symptom onset or imaging findings. So yeah, it would require some preliminary studies.
Yeah, I brought this up because I have been working with colleagues in Germany and Wales on such a simple whole blood assay. I hope it's going to be published soon. It's under review right now, and I could share the details if that could help your studies to compare unstimulated and Aspergillus antigen stimulated protocols in a quite simple fashion.
That'd be great. I'll put my email in the chat box. Thank you.
Great. So David Andes has a question in the chat box to Julie. Julie, do you find single transcripts can differentiate, or do you need to multiplex? And also, do you find differences among candidate species in your transcriptomes?
Yeah, so those are great questions. So between, as far as specifically looking at differences between species, there are very minor differences between the species, but not enough that it's been statistically kind of significant. And so we haven't found significant variation between. And I think that might be one of the benefits of it is that this approach will be broadly applicable to multiple different species of Candida. And then for single transcripts, I assume you may be asking about like single cell, like RNA-seq versus multiplex. We haven't done single cell. We've only done the kind of multiplex or bulk RNA-seq for right now. So I don't directly know the answer to your question unless you were trying to go in a different direction.
Can I ask a quick question, John?
Yes, Demetrius.
For Todd, I'm reflecting on the good old days of Transnet network in the early 2000s, and I think you need to reflect how much the field has changed in the last 15, 20 years. Just to let you know, there is a lot of more center effect now of how chemotherapy is done between centers with all this molecular targeted therapy, how the CAR T cell is positioned in the natural history of lymphomas is also under debate. We have a lot of antifungal drugs compared to the early days of Transnet. And I was wondering, beyond the obviously descriptive nature of a new Transnet, how you're going to address center effect and the complexity and heterogeneity of all these risk factors you're going to try to look at.
Yeah, definitely. I think from the standpoint of like truly understanding how the different sort of protocols and utilization of different therapies I think you really have to pair them with a lot of the sort of interesting and unique technologies that the other speakers have talked about. You need a better understanding of what are the sort of immunologic profiles and differences in sort of the, I guess to borrow one of the favorite terms, net state of immunosuppression. How does that vary across these different states and profiles? And so I don't think you honestly can do it purely from an epidemiologic observational process. I think that's where this addition of biobanking of patient specimens has to come in to be able to actually look at finer details of their immune function and see are there differences between these different groups and how things are being done.
So all you need controls basically? Are you going to, is controls an important element of your study? In order to figure this out, I think you need controls.
Yeah, I think you definitely do. And I think that comes back to, then that creates a much larger and more complex study than even what the original Transnet was. I think it's enrollment of uninfected people in addition to infected patients and being able to make this sort of more basic comparison between groups.
Can I make a comment here?
Yeah, Pete.
Yeah, I mean, Demetrius brings up a great point, and I feel like I should speak because I kind of designed Transnet, and its follow-on was OTIP, which is much less well known because it only involved 6 centers, and it was a cohort study, and it involved basically following everybody at each transplant center, and so those 2 studies represent 2 extremes, and I mean, what's needed here is some hybrid where you can take into account the side effect, because the patients at MD Anderson are going to be quite different than the ones at UAB and at other places. And so you have to have controls taken from those sites individually, but you can't put everybody that you transplant into these cohorts. It's just overwhelming. But to do a really good job, it takes time and lots of money, and it's just a huge data collection effort for sure.
So I'm going to go on to the several other questions. Mihalis is asking Shannon, great presentation. Have you exposed your mice to C-dub in the oral mucosa, not IP, to see if the same protective effect might also be seen?
Thanks for that question. It is a really good question. I don't believe that we have exposed the mice in the oral mucosa, but I will say that we have done IV infections with C-dub, and we do see the same protection. And it's sort of a different answer to your question. We don't see protection against vaginitis when we do IP C-dub vaccinations.
Just a quick thing, Shannon, while you're still talking, are all C-dubs created equally? And is this a unique strain that you have? I mean, I've got a patient in the hospital now that we've been treating for a month and a half with C-dub meningitis. Maybe it's less virulent because we can't get rid of it. It's still more virulent. So the question I have is this system specific for your C-dub, or are there other strains that would similarly act that way?
So I actually don't know that much about different strains of C-dub. There's not like a ton of literature on C-dub, but I will say that we see protection with other low virulence Candida species. So it's not just specific to this one strain that we're using. So I hope that kind of answers your question.
No, I think it does. I think you got a strain that's setting something off, and other strains could do that too if it doesn't kill the animal ahead of time.
Yeah,
so
we're just using
you're
C-dub as a
just
tool.
using it as an example of the immunology that it sets off. Yeah. Okay, I have another question from Pete Pappas, and this is to Stephanie.
Let me ask Stephanie. Stephanie, this is Pete Pappas. I was really interested, number one, and impressed that we have an ENT person on the call today. That's a first. You need to join our group.
I'm delighted. Yeah.
Yeah. But there are several of us on here who have really been challenged to perform a prospective clinical trial in acute invasive fungal sinusitis. And maybe because we've been trying to limit it to one group, the Mucorales or the Zygomycetes, et cetera. But how do you think it's best to deal with the heterogeneity? Because you have not just, a host of organisms that cause this, host of underlying disorders, huge anatomic differences, and the impact and really the necessity of surgery, and then the need for antifungal therapy, we presume in most cases. How do you balance all that? That's a lot of questions, a lot of moving parts, a lot of heterogeneity, and it drives clinical trialists wild. It really drives them wild because there's no homogeneous population. I mean, it's really as heterogeneous a population as you can almost find.
That's a great question. In terms of clinically, I don't know if it's right or wrong, but as surgeons, we treat kind of all acute invasive fungal sinusitis the same. We rely on our, experts in infectious diseases or medicine to kind of balance or try to correct any neutropenia or hyperglycemia as able. But in terms of our surgical approach, we cut it out, So I think—
Yeah, we know about you surgeons.
As I'm approaching my research, I think there's a huge need, and like I'm working on a resubmission of an R21. So I want to get some exploratory data, using the nationwide inpatient sample, trying to get the biggest chunk of patients possible and really try to clarify, are we seeing more diabetics? Are there more transplant? Is there a geographic predisposition? Are there social determinants of health, dilapidated housing or poor access to healthcare that we see more cases? Because I don't know. And so I think that will lay the groundwork for potentially future clinical trials. And in terms of collecting our biospecimens, we see a handful of cases a year at Northwestern. I'm one of 5 sinus surgeons, and we'll see a handful a year, but that's it at Northwestern, which is a bigger center in Chicago. And so we want to collect all comers to start with. And I don't know, maybe in a few years I could answer your question better. And I think you've probably thought about it more from a clinical trials perspective than I have at this point, and would love to hear advice as I set things up.
Well, several of us, I think, would like to talk to you. Okay, because I mean, just a lot of people have been thinking about this for a long time. So
can I ask if anybody is already studying AIFS biospecimens?
Not in a clinical trial setting. Not in a clinical trial setting.
Stephanie, this is a very tricky business, as Pete has talked about. It's as heterogeneous as some of the patients are in cancer and how they're treated. This is a very, very heterogeneous group. I just recently saw a lot of patients with COVID and Mucor. And at the end of the day, and this is a big review, I can't even sort out the steroids versus the diabetes versus the COVID. It's kind of all of the above. What I would suggest that you do, if I was thinking about doing it, is start walking before you start running. And I would be very careful about trying to do the diagnostics on this to find out which organisms they actually are. And in this day and age, you've got PCR sequencing of parafilm, all these tissues, even the blood and stuff like that, to really get an idea of the epidemiology of producing this stuff and the technology of diagnosis. The other stuff becomes very, very tricky. I think.
Just if you want to, if there's any help, we have a paper in press about Mucor post-COVID, and 95% of cases are documented, all of them in diabetics and steroids. So, and what of the people don't understand is there are a lot of mimickers of sinusitis in COVID, because you see a lot of ophthalmic vein thrombosis. So you need to really be careful to really go after this. And reflecting what John said, we published our experience at MD Anderson with invasive fungal sinusitis a couple of years ago. I can send you the paper. And what we have in our hospital, just to see the practice variation, we see the ENT people see it within 1 day. So early rhinoscopy within 1 day of the symptoms. And what we found is the only predictor of outcome was leukemia status. It doesn't really matter, because you start antifungal therapy, you do limited surgery, and then if they go to remission, they survive. If they don't, they die. So be very careful about the heterogeneity, just echoing what Pete said. I'll be more than happy. I'm very interested about this topic, and I'll be more than happy to talk to you about the clinical part.
Thank you.
Do we have any other questions from the group? Anybody on site put up either here or on the chat? I think what I'll do is that we're just about almost into Pete's time here. But I just want to go through a couple things since everybody did such a nice presentation, just what I listened to as I'm going through it. And with Sebastian, I really like doing the leukemia model. I think some of the models in Aspergillus are pretty pathetic. You just kill them with the immune suppression and stuff like that. They either die or they don't. So having a disease that's actually more relevant to what we see in the clinics, I think is impressive. Jeff, with the immunology markers, there's people starting to do this, and I'm tired of actually looking at white blood cells and say, okay, that's a risk versus not. We really need not to understand all what happens in the mouse, but we need to understand what happens in the humans, and you do very detailed immunological studies. I think that's great. Julie, I think specimens. We're on this thing. You've got the techniques and stuff like that. You just need to have more specimens. So everybody out there that can have various RNA systems and stuff like that to get to her, she can actually identify things. For Todd, all transplants, I think transplant centers, I would do some preliminary stuff like Pete said, is look at 5, 6 different big transplant centers and kind of get a feel for what they do, because they're not all the same. And so if you have 5 or 6 that you can use as your canary in the coal mine, that might be important, but you better kind of get and understand each one of those centers pretty well. For Shannon, C. dubliensis, low virulence, I think, again, it's probably a strain that allowed you to get this immunology, which is quite interesting. It may not just be dubliensis, but others. For Reba, when I'm going through this, when you're doing these things and making these randomizations and various things, and trying to make predictions on all these monoclonal antibodies that we're doing, I don't even want to think how I have to do on Thursday when I have a patient that's on crypto and was started on NITNF therapy. But I do want to make sure, I think in your group, that you pay attention to race a little bit. Now, again, there's surely immunology around race. And so doing your things of balancing and stuff, because there are probably certain genetic factors that are fairly important. And finally, for Stephanie, yeah, we struggled with this for a long time. And I think you talked to some of the clinicians about this, the underlying diseases, randomization, the CNS involvement versus not, the surgery and how that's done. And actually the diagnosis is pretty important. And so it's tricky, but it's an area that should be explored. So is there anybody else that's had any other questions from the group? I do want to thank, I was put into this, actually. I wasn't the one who's supposed to do this, but it was enjoyment to actually have to pay attention to each one of your speeches and stuff like your talks, because they're quite interesting. I am very impressed with your passion on this and your ideas on this, and the work that you're actually doing. And hopefully you can interact with a lot of people in the MSG. As was said from Don in the beginning, do not be afraid to reach out to people and ask about questions, interactions, and stuff like that. The fungal community is not a place that if you reach out, they're going to bite you. They're actually probably going to embrace you. So I encourage you to do that. So I think it's now time, we're at 4:22 or so, trying to stay on time. It's back to Pete and his closing comments.
Right. Great. And thank you, John. Thanks to these presenters. And I'll be very brief. Thank you to everyone today for making this first collaborative effort between NIAID and MSGERC in this venue. I think it's been just really better than I expected in terms of attendance and the content. I'm very enthused by what I heard today. Especially want to thank the younger folks, the newer investigators, the 7 of you who presented here towards the last. I think that we'll do anything we can to encourage you and help you along, because I think God knows the field needs an infusion of new and enthusiastic younger investigators. And you sound like you guys are all getting started off on the right foot and have some great ideas. Would like to thank both the intramural and the extramural investigators who presented today, and of course Donna and Beiying, who helped us hatch this idea and really were the catalyst behind this. And I'll come back to where I started, which is why are we doing this? And I think when Donna and Beiying reached out to us, one of their frustrations was they're not getting enough proposals that are of a clinical nature and or clinical translational nature. And so the main purpose for doing this was to kind of light that path, provide some kind of a spark maybe for some of you to start thinking about how to do this. I think that David especially emphasized persistence. They talked about innovation and kind of understanding the system. That's all part of it. But I thought the persistence and the communication with your colleagues at NIAID in the bacteriology and mycology branch really important. I think they're incredibly available and very, very willing to share their thoughts and ideas. And I think that the other thing that I've taken away, I've known this, but that I would really like to change if I could, is how we utilize and work with our colleagues, our intramural colleagues, the Mihalis Leonikis and Steve Hollins of the world. There's all sorts of potential, especially as a group. I'm talking about really group dynamics now, but the group studies that we do, it frustrates me a bit that we have not been able to lasso them in and get them involved in more of the real basic science questions that could be a part of some of our more clinical projects. And I'd love for us to see that change. So I think this has been successful. I would love to get feedback from those of you who participated today, both as participants and as listeners. Any feedback that you have will be valuable, whether it's good or bad. And so please give us some feedback when you have a moment. I will complete this by just encouraging you to look into attending. We are going to meet as a group November 5th to 8th outside of Albuquerque in a really nice spot about 30 minutes outside of town. It's a great venue. It's a resort type of community, a resort type of venue, and we should have a great lineup. We've got tremendous talks, a great agenda. Those of you who've attended these meetings before, you know how valuable it is to meet face to face, but it's also going to be available as a virtual event as well. So I would encourage everyone to look into it. Come if you can. If you can't be there in person, try to attend virtually, and we'll see. So with that, we'll conclude. Hope to see you in November or sooner. Those of you who would like to reach out to any of us, please do. One of the fruits of this discussion today can simply be, pollinating and developing new relationships and so forth. So I think this has been a great exercise, and I hope that everyone agrees with me in that regard. Again, and thanks to Castle & Associates and to Carolyn Jones for all the effort they've put into this for making this a successful event. With that, I think we can sign off. And thank you all. Hope you have just a tremendous day and a tremendous week, and we'll talk soon. A survey is being sent out, so please complete it. John, thank you for emceeing. Tom, thank you for emceeing. Thank you, guys.
Thanks everyone for your attendance. This session will now close.
Thanks all.